Hello everyone, welcome to this session. My name is Kritika Jha and we are here to discuss about the solutions of the botany portions of your recent happened grand test 2. Okay, so in the question paper you may see the biology section as a whole, like botany, zoology are mixed, but if we are discussing solutions, we have divided into botany solutions in a different session and zoology solutions in a different session. So I am here to discuss about the botany questions with you. Okay, so we will start. So the first botany question is your question number 2 in the question paper. The question is saying, which hormone is responsible for horizontal growth, swelling of axis and apical hook formation in dicot seed, which is actually triple response growth. These three developments are in together called triple response growth. Remember, triple response growth is shown by the phytohormone called ethylene. Okay, 3 is your answer. Ethylene does that. Ethylene helps in the horizontal growth. It also helps in the swelling of axis and apical hook formation in the development of dicot seed. Okay, ethylene is your answer. Okay. The next botany question is your question number 3, which is asking, a few ions or molecules are transported across the membrane against their concentration gradient is called. So when any ion or any molecule is transferred or transported against their concentration gradient, against means from low to high concentration, that is your against the concentration gradient, and that kind of transport is called active transport. Okay, second is your answer. Next question. The next botany one is your 5th question. Pollen grains are light and non-sticky. Single ovule in an ovary. Large feathery stigma are characteristics of. So all these characteristics of pollen are specifically found in a particular type of flower. Okay, a particular type of flower have light and non-sticky pollen. It will have only single ovule in that ovary, and it will also have a large feathery stigma. All of these characteristics are found in the pollen grains of flower which are pollinated by wind. Wind pollinated flowers or anemophilous flowers, the option number 1. Okay, anemophilous flowers are those flowers in which the pollination is done by wind, and if pollination is done by wind, the pollen grain has to be very light so that it can easily be carried by wind. It should be non-sticky. The ovule is only one in each ovary, and the stigma is also large and feathery. Why is it large and feathery? To trap the pollen grains in the wind. Okay, so these all are the characteristics of anemophilous flowers. Your option number 1 is your correct answer. The next is the question number 7. So in tissue culture experiments, medulla cells of a stem are kept in culture medium to form callus, and they undergo phenomena known as. So here there are some technical terms, let's understand. So this is a tissue culture experiment. What do we do in tissue culture experiment? We mostly try to grow tissue in a petri dish. We try to differentiate cells. We increase the cell number artificially in the lab in a petri dish. That is your tissue culture experiments. So in this tissue culture experiment, medulla cells of stem. What is medulla cells of stem? Which cells is it talking about? Medulla cells you can understand by medullary rays. I hope you have heard about this. Medullary rays. So stems, dicot stems specifically, have medullary rays, and these medullary rays are parenchymatous, right? They are of parenchymatous origin. Means they are permanent cells, right? Parenchyma is a permanent cell. It has lost the ability of division. Now this medullary rays or medulla cells of stem are kept in a culture medium. If it is kept in a culture medium to form a callus. What is callus? Callus means undifferentiated tissue. undifferentiated mass of cells or tissue. Undifferentiated mass of cells or tissue. Okay, means a lot of group of cells are just present there to form a tissue, but they have not attained any particular structure. They have not formed any well defined structure. It's just a group of cells in a mass. That is your callus. So this callus, when it is formed, when it is continuously dividing, when a cell is continuously dividing, then only it will form a large mass of cells. So when a cell is continuously dividing, what kind of cell is that? Meristematic cell. Am I correct? So see, the medullary cells of the stem is converting into callus, means meristematic cell. So what is the phenomena when a permanent cell which has lost the ability of division is changing into meristematic cell which has the ability of division? This change or permanent cell to meristematic change is called de-differentiation. Okay, so the answer is second one. De-differentiation is your answer. Okay. Okay. The next question is your question number 12. How many ATP and NADPH plus H plus are utilized during production of one net sucrose molecule through Calvin cycle? Be very careful. The key word here asking is you sucrose, not glucose. Sucrose is made up of what? Sucrose is made up of glucose plus fructose. Okay, glucose and fructose, these two are isomers, structural isomers, means their chemical formula is same. Okay, their chemical formula is same, C6H12O6, both for glucose and sucrose, but their structure is a little bit different. It's just based on the difference of the position of the atoms. So that is why if glucose and fructose are together, it is basically the same. Okay, so we can say that it is actually two glucose molecules. Okay, because they are isomers. They are isomers, right? Their structure is similar. Their chemical formula is their structure is though different. Their chemical formula is same. So we can say that ultimately they are forming two glucose molecule only. Okay. Now, Calvin cycle. So first of all, you have to know how many ATP and NADPH are used in Calvin cycle. So let's see. When one CO2 is fixed, when one CO2 enters the Calvin cycle, it is fixed, right? How many ATP and NADPH is used for that? 3 ATP and 2 NADPH. 3 ATP and 2 NADPH is used for one CO2 fixation by one turn of Calvin cycle. But if we want to make one glucose molecule, if we want to make one glucose which is C6H12O6, how many carbons are there? 6 carbons, which means how many carbon dioxide molecules we will need? 6 carbon dioxide molecules we will need, which means how many Calvin cycles turn it will be? 6 cycles will be there, right? 6 Calvin cycles will be there, and in 6 cycles, how many ATP and NADPH is required? 6 into 3, 18 ATP. 6 into 2, 12 NADPH. So for one glucose, we require 18 ATP and 12 NADPH, but here we have sucrose molecule, one sucrose, which means 2 glucose. So for 2 glucose, how many ATP NADPH will be required? 18 into 2, 36 ATP. Sorry, 18 into 3, I am so sorry, 18 into 3, 36 ATP, and 18 into 2, sorry, 18 into 3, 36 ATPs are required. Okay, my bad, yeah. So in one glucose molecules, 18 ATP and 12 NADPH is required. So if we have 2 glucose molecules, we just multiply it by 2. 18 into 2, 36 ATP, 12 into 2, 24 NADPH. So your answer is your option number 3. For 1 sucrose, basically glucose plus fructose, but since they are same, we consider it as 2 glucose. So for 1 sucrose basically means 2 glucose molecules, we will need 36 ATP and 24 NADPH plus H plus. Okay. The next question. So the question is, we have to identify the incorrect statement. The question number 16, okay, we have to identify the incorrect statement regarding polygenic inheritance. So what is polygenic inheritance? Polygenic inheritance is the expression of phenotypic characters or expression of a character or a phenotype which is controlled by more than 2 genes. Okay. Your answer for this question is 1. The first one is incorrect. The first option is saying a single gene affecting multiple phenotypic expressions. A single gene, when it affects multiple phenotypic expression, is not polygenic inheritance. It's what? It's pleiotropy or pleiotropic gene. Okay? When a single gene affects more than one phenotype, it's pleiotropy. Polygenic means expression of phenotypic characters is controlled by more than 2 genes. When one character is controlled by more than 2 genes, that is polygenic inheritance. Polygenic inheritance is also affected by the involved correct, and it is not a Mendelian inheritance. It is a non-Mendelian, means basically it's a non-Mendelian inheritance. Okay, so first option is wrong about polygenic inheritance. Next, so it's an assertion based question. So assertion is saying dorsiventral leaf is a bifacial leaf. What is bifacial leaf? Bifacial means 2 faces, 2 different faces, 2 different structures of that. Okay. The reason for that is that in dorsiventral leaf, the mesophyll is differentiated into upper palisade and lower spongy parenchyma. So both is correct, and this reason is also correctly explaining the assertion. So what is it saying exactly? So this, for example, is your dorsiventral leaf. This is your upper epidermis, this is your lower epidermis. Now you know that in dorsiventral or dicot leaf, the mesophyll cells, they are divided into 2 types. The upper part have long parallel elongated cells, which is your palisade parenchyma, right? But the lower part have spherical, large or oval, what? Spongy parenchyma. Okay? So these are the 2 types of mesophyll cells of dorsiventral leaf. So basically it is 2 face, right? One upper face, one lower face. So that is why it is called bifacial, due to the different mesophyll cells. Okay, so that is why your answer is first one. Okay. Next. So during cell cycle, okay, during cell cycle, the centriole duplication occurs in. So centriole duplication. Centriole is duplicating in the cytoplasm in S phase. But remember, only in animal cells, because centriole or centrosome is a organelle only found in animal cell, not in plant cell. So in animal cell, during S phase, DNA replication happens inside nucleus. Also centriole duplication happens in cytoplasm. Okay, so fourth is your correct answer. Next is your question number 22. Solanaceae and Malvaceae, these are the two different families, resemble each other in the following character. So Solanaceae means it's talking about potato family. Potato, brinjal, tomato, chili, datura, all of these comes under Solanaceae family. Malvaceae means it's talking about hibiscus. Hibiscus family, China rose family. So basically we have to see how these two families are resembling in each other. Gamopetalis, gamopetalis means all the petals are fused together. Gamopetalis corolla only seen in Solanaceae, not in Malvaceae. Okay. Polypetalis means all the petals are separate, they are not fused. Polypetalis is only seen in Malvaceae, not in Solanaceae. Monoadelphous condition. What is monoadelphous condition? So we have a stamen, right? We have a stamen. Stamen has two parts. This is your filament and this is your anther, correct? So when all the filaments, I am saying all the filaments, if there are more than one, if all the filaments of a flower, of all the filaments of the stamen of the flower, if they are fused in one bundle, then it is called monoadelphous condition. Okay. So in China rose specifically, or we could say Malvaceae family. In China rose, there are a lot of stamens. All the filament of these stamens, they are fused together in one single bundle, one single tube. Okay, that is your monoadelphous condition. It is only seen in Malvaceae. Solanaceae, their stamens are separate. The last one is epipetalis condition. This epipetalis condition means the stamens are fused with the petals. When stamens are fused with the petals, that is called epipetalis condition, and it is seen in both of them, Solanaceae and Malvaceae. In Solanaceae, all the stamens are separate, but all these separate stamens are fused separately with the petals. Okay, that is one kind. In Malvaceae, there is monoadelphous condition. All the filaments are fused in one single bundle, and that one single bundle is also fused with the petals. The lower part of that single bundle is also attached with the petals. That is why in Malvaceae also we have epipetalis condition. So in both Solanaceae and Malvaceae, we have your epipetalis condition. In Solanaceae, all the free stamens are attached separately to the petals, whereas in Malvaceae, all the filament of the stamens, though they are fused in one bundle, the lower part of that single bundle is fused with the petals. Okay. Next, from the following option, set of plants that show basal placentation. You know what is basal placentation? The placenta arises from the base of the ovary, and there is normally single ovule in that ovary. Okay, that is your basal placentation. In NCERT, the examples are mostly given about sunflower, right? It's mostly talking about sunflower. But here, apart from sunflower also, you will have to know some examples. So, your answer for this is third one. Oryza. Oryza is rice. Okay, Oryza, Tridax, Helianthus. Tridax and Helianthus, they come in the sunflower family itself. Okay, Asteraceae family, sunflower family itself. Okay, so basal placentation example apart from sunflower also is Oryza, Tridax, Helianthus. Third is your answer. Okay. Next question is your 25. Question number 25. We have to identify a pair of heterosporous flowering plant. Very important to read the question properly. Heterosporous means two different spores, two different types of spores, one microspores, one megaspores. two different types of spores, heterosporous condition is also seen in some of the pteridophytes, right? Some of the gymnosperms, most of the gymnosperms, right? In some of the pteridophytes, in some of the gymnosperms, in most of the angiosperms, all of the angiosperms basically, we have heterosporous condition. Microspores, megaspores, microspores will develop into male gamete, megaspores will develop into female gamete or gametophyte, right? But here we don't know, or do we know? What is this? Flowering plants. Flowering plants means angiosperms only. So we don't have to select pteridophytes, we don't have to select gymnosperms, we only have to select flowering plants, specifically angiosperms. Okay. So in the whole options we'll have to find the angiosperms. The first one, Salvia selaginella. Salvia and Salvinia, two different things. Salvinia is a pteridophyte, but Salvia is a angiosperm. Okay, so Salvia is a flowering plant and Selaginella is a pteridophyte, so we will not select this option. We will only have to select angiosperms. Salvinia, Salvia, again Salvinia, even though it's a pteridophyte, Salvia is an angiosperm, but still not both of them are angiosperms, so again we will not select. Selaginella brassica, Selaginella pteridophyte, brassica flowering plant, not okay. Last one, Salvia angiosperms, Solanum angiosperms. So both of them are angiosperms. So your answer is 4. Heterosporous flowering plants means heterosporous angiosperms, Salvia and Solanum. Okay. Next question. What is the function of silky hairs in corn cob? What is the silky hairs in corn cob exactly? Silky hairs of the corn cob is actually its stigma and style. Okay. The corn cobs which has silky hairs, which you may have seen while peeling them, is their style and stigma. Why they have these kind of silky feathery style and stigma? To trap the pollen grains, which is traveling with the wind. They are anemophilous. Okay, to trap those pollen grains which is traveling in the wind, these stigma and style, they have hair like feathery like structure. Okay. Next is question number 29. So we have to match the animal with list 1 with their extinction in list 2. So these are animal list which has become extinct now. They are extincted animals, but from where they have extinct is the list 2. So let's see. Though the answer for this question is 4th one. Okay, 4th is your answer. So dodo was originally from Mauritius. It got extinct from Mauritius. Quagga, it got extinct from Africa. It was an African animal. Thylacine was initially found in Australia, from where it got extincted. And Stella sea cow was originally found in Russia, from which it also was extincted. So your answer is 4th. Okay. Next question. So again we have an assertion reasoning question. So let's see. So assertion is saying gametophytic stage in moss plant is dimorphic. Dimorphic means two morphologies. Di means two, morphic means morphologies. So two different morphologies, two different structures. So gametophytic stage of moss plant is having two different morphologies. Is it? Yes, we have two different morphologies for the gametophyte of moss, right? The juvenile filamentous stage is called protonema, correct? And the adult leafy stages are seen in the gametophyte of moss. Yes. So gametophyte of moss, they are divided into two life stages, juvenile and adult or leafy. So juvenile is a green, creeping, filamentous, photosynthetic gametophyte, whereas the leafy stage is also the mature gametophyte of moss. Okay, so these are both the different morphology or dimorphic phases of the gametophytic stage. So what will be the answer? The answer will be 1. Both A and R is correct, and R is the correct explanation of A. Okay. Next question is your question number 35. So phytohormone that helps in development of adventitious shoot and lateral shoot growth is? So the phytohormones which helps in the adventitious shoot and lateral shoot growth is cytokinin. Cytokinin is the one which will help in suppressing the apical dominance. It overcomes the apical dominance that allows the lateral shoot growth, okay? And it also allows development of adventitious root, okay? All because it can overcome the apical dominance of auxin, okay? So cytokinin is your answer. Next, verification of chromosomal theory of inheritance. Verification means experimental verification or proof, right? So this verification of chromosomal theory of inheritance was done by T.H. Morgan. The answer is 2. Morgan did his studies on Drosophila to experimentally prove the chromosomal theory of inheritance. Okay. Next is question number 37. So this is a diagram, okay. This diagram is showing the feeding interactions among the various species from A to I. A, B, C, D, E, F, G, H, I, all these species and their feeding interactions. Basically this is a food web. Okay, so now there are some options regarding this food web. So we have to choose which statement is correct. So first of all, they are at different trophic levels. See, trophic levels are mentioned. This A, B, C is at trophic level 1. F, D, E is at trophic level 2. G, H is at trophic level 3 and I is at trophic level 4. So we know that the lowest trophic level is your producers. Am I correct? The plants. So A, B, C, they are plants or producers. Second, those who eat plants, right? F is eating A, D is eating B, E is eating C. All of these are eating plants. So the level 2 is your, or trophic level 2 is your primary consumers. Those consumers who eat producers like herbivores mostly. Now, the trophic level 3, in trophic level 3 we have G and H, and this G and H is eating your primary consumers. This G is eating F, D, this H is eating E, right? And D also, correct? So it's eating the primary consumers. So the organisms who eat primary consumers are called secondary consumers. Mostly they are carnivores. Okay? Now last trophic level, the I, which is who is I eating? G. G is eaten by the I, right? So the organisms or the consumers who eat secondary consumers is called tertiary consumers or final consumers. Okay, they are the highest trophic level. Now let's see the options. Species G is climax consumer. Climax means top level consumer. So species G is top level? No. Species I is top level, not G. G is secondary consumer. Second option, species B is a herbivore. B is a producer, it's a plant, not a herbivore, no. Species H is an omnivore. Now what is omnivore? Omnivore is those organisms who will eat everything. It will eat plant, it will eat animal also. Everything it will be eaten by omnivore. Now species H, so this is here where species H is. Species H is eating D, species H is eating E. D and E are primary consumers. So is species H eating any plant? No, right? So it's not an omnivore because it is only eating the primary consumers. It is only eating animals. It is not eating plants. So it's not an omnivore. So what is the answer? The fourth one. Species D is an omnivore. So this D, species D is eating B and C, which are plants. Species D is eating E, which is an animal. So it is eating plant also, it is eating animal also. It's a omnivore. So the answer is D. Okay. Next. So again an assertion reasoning question. So in glycolysis, there is net gain of 2 ATP molecules from SLP reaction. What is SLP? SLP means substrate level phosphorylation. So the here I have given the chart for glycolysis for your understanding. So there is net gain of 2 ATP from SLP reactions. One ATP is gained here, another ATP is gained here. These are both substrate level phosphorylation which helps in the formation of ATP. Okay, so yes, total how many ATPs are formed? 4 ATPs, right? Because we have 2 molecules of everything. So if one molecule of this is formed, is converting into one molecule of phosphoglyceric acid, means if one molecule of 1,3-bis phosphoglyceric acid is converting into one molecule of 3-phosphoglyceric acid, it requires 1 ADP, means it forms 1 ATP. So if there are 2 molecules, it will form 2 ATP. So 2 ATP here, 2 ATP here. We gain 4 ATP, but here at the starting of the stage, 2 ATPs are used also. So if 2 ATPs are used, then what will be the net gain? Net gain will be 4 minus 2, 2 ATP. So yes, net gain is 2 ATP, which is correct. So reason is in glycolysis 2 ATP molecules are used in initiation or activation phase, which is also correct. Basically what we have discussed. So the answer is 1. Both A and R is true and R is the correct explanation. Okay. Next question. For the first time, which is the question number 44, for the first time, proof that DNA acts as genetic material comes from the work of. So there were a lot of scientists who did a lot of experiments to find out which is the genetic material, right? First one was Griffith. He did the transforming principle, but he didn't tell that it was DNA which is getting transferred. He just told that, okay, something is getting transferred from the heat-killed S strain to the live R strain, which makes it virulent, okay, and makes the mice die. He didn't know it was DNA. So who came and told that it was DNA which is doing this? For the first time, it was second option, Avery, MacLeod and McCarthy. Avery, MacLeod and McCarthy, they just repeated the Griffith experiment. The change what they did was that when they were doing the Griffith experiment, so what was the final Griffith experiment? That heat killed S strain plus live R strain. When it was injected in mice died. Why? Because something from this killed S strain was getting transferred into live R strain, which made the R strain virulent. Okay. So how did Avery, MacLeod and McCarthy found out, okay, it was DNA which is transferred? So they treated this heat killed S strain, okay, they treated this heat killed S strain first with proteases. Proteases is the one which denatures the protein or destroys the protein. They saw that despite being treated by proteases, the mice is still dying. Means it is not, protein is the not the biomolecule which is getting transferred. Next they treated it with RNAs. RNAs is the one which denatures the RNA. Same result, again the mice died, means RNA was also not the one which is getting transferred. When they added DNAs, only then they found that, okay, now mice is living, which means DNA which is destroying the DNAs which is destroying the DNA, it destroyed the DNA of S strain, that is why it was not able to transfer into live R strain, and that is why it was not able to make it virulent, and that's why mice lived. So that's how they proved that, okay, DNA is the one which is getting transferred. Okay, so Avery, MacLeod and McCarthy was the one who did that for the first time. Next, which of the molecule cannot absorb a photon? Which cannot absorb a photon? Chlorophyll A of PS2, it can. It's a pigment, it can absorb a photon. Chlorophyll B, yes it can. Chlorophyll A of PS1, yes it can. All the pigment molecules, they can absorb photon. But the last one, what is PC? PC is for plastocyanin. PC stands for plastocyanin, which is a part of electron transport system, okay, and it is not absorbing photon, it carries electron, it cannot absorb photon, okay. So fourth is your answer. Next question, which is question number 49. From the following, we have to identify haploid, diploid, triploid, nutritive tissues respectively. We have to identify haploid, diploid, triploid, nutritive tissues. Nutritive tissues means the tissues which is providing nutrition. So first of all, let's see which is providing nutrition. Nucellus. It provides nutrition? Yes, nucellus provides nutrition to the developing female gametophyte of embryo sac or embryo sac, right? So nucellus will provide the nutrition. Endosperm, yes, it provides nutrition to the developing embryo. Again endosperm, yes. Egg cell is a gamete, it does not provide nutrition, so this will not be considered. Cell of endothecium in anther, which is the tissue which is providing nutrition to the developing pollen grains. It is the tapetum, the innermost layer, not the endothecium. Endothecium helps in dehiscence. Okay, so this is not also considered. So out of these three nutritive tissues, we will have to find out which is haploid, diploid or triploid. So nucellus of P ovule. P is a angiosperm. Nucellus is a part of ovule. Nucellus is a part of ovule and it's a angiosperm, so it will be diploid. Endosperm of Pinus. Now Pinus is a gymnosperm. Okay, remember Pinus is a gymnosperm and endosperm of gymnosperms are haploid. Okay, so endosperms of Pinus or gymnosperms are haploid. Endosperm of rice. Rice is a angiosperm and in angiosperm we have double fertilization. So endosperm of rice will be triploid. So haploid, diploid and triploid respectively means first we have to select the haploid. So B, diploid is A and triploid is C. So BAC, the answer is second. Okay. Next, element that is component of cytochrome C oxidase complex is. So element which is found in cytochrome C oxidase. Cytochrome C oxidase is your complex 4 of electron transport system, and inside that complex, which component is found? Copper. Okay, copper A, copper B, right? So copper is your answer. Next, the biomass available for consumption by herbivores and the decomposers is called net primary productivity. The answer is 1. Whatever biomass the plant is forming by the help of photosynthesis, that is called gross primary productivity. Not all of them is available for the consumption because plant also uses some of the biomass during respiration process. So if we subtract gross primary productivity and we subtract respiration loss from it, then we will get net primary productivity, and this net primary productivity is the one which is available for consumption. Okay, the answer is 1. Next question, the stage in meiosis where each chromosome shows one chromatid. Each chromosome is showing one chromatid like this. This is your final stage, means telophase 2. Okay, correct, telophase 2. Next, Gauss principle of competitive exclusion states that. So what does the Gauss principle of competitive exclusion states? They state, the answer is third one, that no two species competing for the limiting resources can coexist indefinitely in the same ecological niche. In a habitat, in an ecological niche, we always have a limited resources, and if there are two or more species competing for that same limited resources, they cannot exist together for a very long time. Why? Because there will be competition between them. So more fittest, more stronger species will can fight for more resources easily and can ultimately cause the extinction of the less fittest species. Okay, that is your competitive exclusion. No two species competing for the same limiting resources can coexist indefinitely in the same ecological niche. Okay. Next, which of the following is not translated During eukaryotic translation means conversion of mRNA to protein. So which part of mRNA is not translated, not converted into protein? So in mRNA, we have a coding region. For example, this is the coding region for which we have to make main coding region. Okay, main coding region for which we need protein. We also have UTRs, untranslated region. These UTRs, they don't get converted into protein, they just help in the translation, effective translation process. And then in the 5' to 3', right, in the 5' region we have a cap, methyl guanine cap, and in the 3' region we have what? Poly A tail. So what is only translated? Only this is converted into protein, not UTRs or cap or poly A tail. Okay, so which of the following is not translated? Cap, tail and UTR. UTR mean untranslated regions are not translated. 1, 2 and 4. The answer is 3. Okay. Next question. If a color-blind man marries a woman who is homozygous for normal color vision, the probability of their sons being color blind is. So what we have? Color blindness. Color blindness, first of all, is what kind of disease? It's an X-linked recessive disease. So color blind man is there. For color blind man, we only have one X. It will carry the C, means colorblind gene. So this is a colorblind man. And he marries a normal woman. Okay, homozygous normal woman means it's XX. She is not a carrier. She is homozygous normal means she doesn't even have a carrier gene. So this colorblind man marries a normal woman. So what is the probability of their sons getting color blindness? The answer is 0%. Let's make a Punnett square. So X, C, Y. These are the two sex gametes of male. Correct? For the female, we have X and X. So X, XC, a female carrier. X, XC, again a female but only carrier. X, Y, normal male, X, Y, normal male. So all the sons are normal only. It's not color blindness. So for son to have color blindness, mother at least has to be carrier, because in sons the X chromosome is coming from the mother. Okay. So in this case, when the mother is normal without even a carrier gene, the sons will never have color blindness, 0%. Okay. Next question. Which of the following is very important parameter chosen for assessing impacts of any biotic or abiotic factors on population growth in exponential growth model? Okay. So the parameter which is chosen to study, okay, this is chosen for assessing impacts of whatever biotic and abiotic factors is there on the population growth in exponential growth model. So the answer is R. The first one, R value. Okay, R stands for, or R value stands for intrinsic growth of natural increase. Intrinsic growth of natural increase, which is equals to B minus, this intrinsic growth of natural increase equals to B minus D. B means birth rate minus death rate. Okay. So this R, birth rate minus death rate, is very important parameter, because biotic and abiotic factors can easily affect birth rate or death rate in a population growth. For example, there is a population in which birth is also happening, death is also happening. But for now, let's suppose abiotic factors, like there is a drought, or there is a volcano, or there is an earthquake. It can lead to deaths of many individuals, right? So population number will, or population size will decrease, correct? So abiotic factor is affecting this R value, correct, population size is decreasing. If we consider biotic one, for example, in that area there are predators which is eating the animals, which is eating the individuals, then also population size will be affected, right? So this R value is the best parameter to assess the impact of biotic or abiotic factor on a population number or population density. Okay. Next question. So there are two statements. Primary xylem is differentiated into protoxylem and metaxylem. Which is correct or incorrect? It's correct. Primary xylem is differentiated into protoxylem and metaxylem. Protoxylem is the first appearing primary xylem. Metaxylem is the later appearing primary xylem. Okay. Next is secondary xylem. Secondary xylem is not differentiated into protoxylem and metaxylem. Yes. Secondary xylem is not differentiated into protoxylem and metaxylem. Only primary xylem is differentiated into proto and metaxylem. So here, what is the answer? 1. Both statements are correct. Okay. Next question. Incorrect statement. We have to identify the incorrect statement. So the first is, during meiosis, the chromosomes are fully condensed in diplotin. This is wrong. Okay, your answer is the option number 1 because this is wrong. During diplotin, what happens? The dissolution of synaptonormal complex. Okay, in the prophase 1 of meiosis 1, we have 5 substages: lactotin, zygotine, pachytine, diplotin and diakinesis. And diplotin, what happens after recombination? Pachytine is the where recombination is happening. After pachytine comes diplotin. So after recombination, the synaptonormal complex or the synapse which is formed will start to dissolve, and it leads to little bit decondensation of chromosomes also. So in diplotin, they are not fully condensed, but last stage, diakinesis, which is the end of prophase 1, is the stage where the chromosomes are fully condensed. Okay, so it should not be diplotin, it should be diakinesis. Okay, so first is the incorrect one. Okay, next question. Species diversity generally increases as one proceeds from. The answer is third, high altitude to low altitude, and from high latitude to low latitudes. So let's suppose this is your globe. Okay, this is your earth, this is your globe. This is the equator, 0 degree. This is your Tropic of Cancer, 23 degree north. This is your Tropic of Capricorn, 23 degree south. And these are your further latitudes. Okay. So this is your higher latitude, right? This is your 0 degree is your lower latitude. Again, this is higher latitude. This is your lower latitude. Okay. So if we are talking in respect to latitudes, where is the most species found? Near equator, right? Highest diversity is found in equator regions, right? Species diversity is found in equator region. So 0 degree or lower. Okay. So as you go from higher to lower, means as you come from the height from the earth poles towards the equator, the number of species will increase, species diversity will increase. Okay. So as you come from higher to lower latitudes, species diversity increase. Altitude means height, same thing. Poles are at high altitude, equator is at low altitude. So again, when you come from high to low altitude, species diversity will increase. So both in case of altitudes and latitudes, higher is where the species diversity is less, lower is where the species diversity is more. So if you come from higher altitude to lower altitude or higher latitude to lower latitude, species diversity will increase. Okay. Next question, again an assertion reasoning. Gymnosperms show direct pollination. Reason is, in gymnosperms ovary is absent. So what is the answer? The answer is fourth. Both assertion and reason are true, and reason is the correct explanation. So in gymnosperms we don't have ovary, we only have ovule. This is the ovule which is surrounded by a single integument, right? And this is the micropyle area, okay, which is open. And there is no external anything, there is no ovary here. This is just ovule and integument in gymnosperms, right? Ovary is absent. And that is why, since ovary is absent, there is no space for pollen tube formation, so that the pollen grains has to reach here directly, okay, directly to the mouth of ovule, so it can directly enter, okay. So that is why in gymnosperms we have direct pollination, pollen grain reaching the mouth of the ovule, okay, because they don't have a ovary. So both assertion reasons are true, and reason is the correct explanation of A, okay. Next question, we have to identify true and false with respect to genetic diversity, okay. So genetic diversity is a diversity shown by single species at genetic level, which is true. Yes, genetic diversity is the diversity shown by single species, individuals of a single species at a genetic level. It is defined as in the total genetic information contained in all individuals of community is wrong. See, when you consider all individuals in a community, it means it's talking about more than one species, but genetic diversity is only for single species, okay. So remember, when we are talking about individuals of a community, means we are talking about individuals of more than one species. So this is becoming species diversity, not genetic diversity, okay. Genetic diversity is only for a single species in an area. Now, third one, C, Raufolfa vomitoria in Himalayan ranges show variation in the terms of concentration of active chemical racapine, which is again true. This is the example of genetic diversity. And the last one, India has more than 50,000 genetically different strains of mango, false. India has more than 50,000 genetically strains of rice. For mango, it's only about 1,000, okay. So what is the answer? The answer is 2. True, false, okay. Next question. So which of the following statements is not correct regarding chemiosmosis? Chemiosmosis is seen during light reaction. Chemiosmosis happens during light reaction for ATP production. Chemiosmosis is strictly related to ATP production during light reaction. So first, which is the following is not correct? The answer is your first one. Why? So it's saying this chemiosmosis requires a proton pump. Yes, it requires a proton pump. Membrane, thylakoid membrane, where the process is happening. Proton gradient, yes, chemiosmosis is the name of proton gradient formation. NADP synthase enzyme, no, not NADP synthase. What do we need? ATP synthase. Because chemiosmosis is strictly related to ATP synthesis. So NADP is not considered in chemiosmosis, okay. So it's not NADP synthase enzyme, it's ATP synthase enzyme. So that's why the first one itself is your wrong, okay. First is your answer. Next, okay, we have to match the list of the prophases, different substages of prophase with what exactly happens in them. The answer for this one is third one, okay. So zygotine is the second stage of prophase 1. So what happens in zygotine? Synapse formation, synapses, or synaptonomal complex formation starts in zygotine. Diakinesis is the last stage of prophase 1. What will happen there? Terminalization of chiasmata. Chiasmata is the X-shaped structure where the homologous chromosomes have done recombination, okay. For example, if this is your one chromosome, this is your another chromosome, okay. For example, this is the place where recombination can happen. So here they have an X-shaped structure, okay, which is called chiasmata. So this chiasmata is terminalized, means it reaches the terminal ends in your diakinesis, okay. Diplotin, dissolution of synaptonomal complex happens in diplotin, and pachytene is the stage where main recombination happens with the help of recombinase enzyme. Okay, so third is your answer. Okay, next question, again an assertion reasoning. So possibility of a female becoming hemophilic is extremely rare. Why? Mother of such female has to be at least carrier and father should be hemophilic, which is unviable in later stages of life. So hemophilia again is a X-linked recessive disease. Okay, so if it's an X-linked recessive disease, for female to carry this hemophilia, female to suffer from hemophilia, both the X chromosome has to be HH, right? And how will it happen? This female child will need father and a mother. Father will also give one X chromosome. So one X chromosome from father has to be XH, means hemophiliac patient father. Father will be hemophilic. Now another X chromosome is coming from mother, so mother at least has to be carrier. Okay, so mother has to be at least carrier and father has to be a hemophilic patient, then only there is a chance that the female child can become hemophilia, which is very rare because hemophilic patients, they don't survive for a very long time. Okay, hemophilia is the condition in which if you get a if you get a cut in your body somewhere, the blood will not clot because your clotting mechanism is basically not working. Okay, so blood will keep on flowing and you will not even realize. Okay, and that's why they die. So the patients who are suffering from hemophilia, they don't live that much, at least until the age where they will get married and have children. It's not very common. Okay, that's why females suffering from hemophilia are extremely rare. Okay, so both the assertion reasons are true and R is the correct explanation. Okay, next question. We have to identify the correct statements. First statement is root hairs are multicellular extension of epidermal cells. It's not. It's wrong. They are unicellular extension, not multicellular, unicellular extensions of epidermal cells. Next one. In the leaf, the ground tissue system is represented by mesophyll. Yes, the ground tissue system of leaf is called mesophyll. Third one, radial vascular bundles are always closed. So what is radial vascular bundles? Radial vascular bundles is seen in roots, okay, where xylem and phloem, they are on different radiuses. For example, this X is actually your xylem and this P is your phloem. Okay, so xylem and phloem, they are on different radiuses, that is your radial vascular bundle seen in roots. Why is it saying it's closed? So since they are on different radiuses, this xylem and phloem, they are on different radiuses, there is no vascular cambium in between them. They are not open, right? Like there is nothing to present between them, right? They are not open, there is no vascular cambium between them. So we can consider that, okay, they are closed. So yes, in a way we can say that radial vascular bundles are always closed. Next, in primary root, the outermost layer is epiblema. Yes, the epidermal, outermost epidermis of root has a name called epiblema. Okay. Last one, the parenchyma cells which lie between xylem and phloem strands in roots form conjunctive tissues. Yes. Okay. So these parenchyma cells here, the parenchyma cells present between xylem and phloem of roots. Between xylem and phloem of roots, they are called conjunctive Okay, so the parenchyma cells found between the xylem and phloem bundles, they are called conjunctive tissue. Okay, which is correct. So which are the correct statements here? Second option, B, C, D, E. Okay. Next, ribozyme 23S should be R, okay. Ribozyme 23S rRNA, okay, not same rRNA, 23S rRNA, okay, is present in which subunit of ribosome? It's present in 50S subunit of ribosome. 50S is the larger subunit of the prokaryotic ribosomes, right? 50S and 30S make the 70S. 50S has this ribozyme 23S rRNA. What is its function? It has a catalytic activity. It helps in the peptide bond formation. In 50S RNA, when amino acid comes, peptide bond is formed between those two amino acids. That peptide bond formation is catalyzed by this 23S rRNA ribozyme, okay. So that is why it is found in 50S subunit of ribosome. Next, which of the following statements about honeybees is incorrect? Let's see. Males do not have a father, thus cannot have a sons, but have a grandfather and have grandsons. This is correct. Next, drones are fertile even though they are haploid. Yes, drones are the male bees. They are haploid, but they are still fertile. Workers are sterile because they are haploid? No. Gametes are produced by mitosis in drones. Yes, drones are haploid, right, male. So if they have to produce gametes, they will do by mitosis. But the third one is wrong. Why? Workers are sterile because they are haploid? No. Workers are female bees only. Workers are female diploid bees, okay, but they are sterile, means they cannot reproduce, okay. It's not because they are haploid, but it's the nature. It's the nature which has decided that that female sterile bees, diploid bees are called workers, okay. There is a hierarchy in the honeybees, okay. Next. Identify the incorrect regarding succinic dehydrogenase enzyme. Succinic dehydrogenase enzyme. You may have heard this succinic dehydrogenase in the Krebs cycle as well as in ETS or electron transport system. The answer for this is second one, B only is your incorrect. So let's see. Succinic dehydrogenase, or it can also be called as succinate dehydrogenase, it is bound to the inner mitochondrial membrane. Yes. This succinic dehydrogenase in ETS, in electron transport system, it forms your complex number 2. It is found in the inner mitochondrial membrane and it's an immobile. So this is true. Malate is competitive inhibitor of succinic dehydrogenase. Inhibitor or anything. Malate is just formed in the Krebs cycle in the later stages, right? Malate is an intermediate formed in the later stages of Krebs cycle. It's not an inhibitor, okay. C. It belongs to class 1 enzymes according to IUB system of enzyme classification. According to IUB system of enzyme classification, class 1 enzymes are oxidoreductase enzyme. All the oxidation reduction process done by the enzyme, those enzymes which do the oxidation reduction process are called oxidoreductase and they are in class 1. So this succinate dehydrogenase, what it does? It does an oxidation reduction reaction, right? During Krebs cycle, it converts succinate to fumarate, or should I say it oxidizes succinate to fumarate. This is a oxidoreductase reaction, right? So yes, it belongs to class 1. Last one, it also acts as a complex 2 in electron transport system. Yes, so the only incorrect one is B. Okay. Next question. Okay, in a plant, the height is controlled by 3 genes. Minimum height is 30 centimeter, maximum height is 60 centimeter. Then what is the height of genotype? Capital A, small a, capital B, small b, capital C, small c. So height, plant height, if it is controlled by 3 genes, means it's talking about polygenic inheritance, right? And what happens in polygenic inheritance? When one character or one phenotype is controlled by more than one gene, then it is your polygenic inheritance. And in polygenic inheritance, you may have heard a word, the alleles effect is additive. Okay, they are additive effect, means each allele will have a additive effect. Okay, so minimum height will happen with which kind of genotype? Small a, small b, small c will give you the minimum height, which is your 30 centimeter. But it's a additive effect, means all the alleles, if you add a plus b plus b plus c will equal to 30 centimeter. Okay, this is your additive effect. This is the meaning of additive effect. So just simplify it. 2a plus 2b plus 2c is 30 centimeter. So if you take 2 in common, a plus b plus c equals to 30. So what will be a plus small b plus small c? It will be 15 centimeter. Okay. Now maximum height is 60. So for maximum height, the genotype will be capital A, capital B, capital B, capital C, capital C. It is your 60 centimeter. So same as that, A plus capital B plus capital B plus capital C, it gives you 60. 2 capital A is 2 capital B is 2 capital C gives you 60. So 2 common, capital A plus capital B plus capital C, 60. So capital A plus capital B plus capital C is what? 30 centimeter, right? Now what we have to find? We have to find the height for this hybrid, right? So for hybrid conditions, what will happen? Capital A, capital B, small b, capital C, small c. You have to add additive effect, capital A plus small a plus capital B plus small b plus capital C plus small c. Can we rearrange it? A plus B plus C plus small a plus small b plus small c, right? So we have calculated it separately. Capital A plus capital B plus capital C is 30 plus small a plus small b plus small c is 15. So what is the final answer? 45. Okay, so the height for this hybrid is what? 45 centimeter. And this is how you will do these kind of questions. Okay. Next question. So there are some compounds or intermediates, should I say, is given in list 1, and in list 2 numbers are given. Basically they are asking you number of carbons. You have to match the molecule with the number of carbons found in them. Okay, the answer for this is second one. Phosphoglycolate, it's formed during photorespiration. It has 2 carbons. 3-PGA, 3-phosphoglyceric acid, is the first intermediate product of the Calvin cycle, has 3 carbons. Alpha ketoglutaric acid, intermediate in Krebs cycle, has 5 carbons, and isocitric acid or citric acid basically has 6 carbons. Okay, second is your answer. Next, in Fabaceae, the flower is an assertion reasoning. In Fabaceae, the flower is zygomorphic. And the reason is in Fabaceae flower consists of papillonaceous form of corolla. Fabaceae, first of all, you have to understand, means Fabaceae family means Leguminosae family means pea family. It's talking about pea. Pea is zygomorphic. You cannot divide pea into more than one plane. Sorry, what is zygomorphic? Zygomorphic means the flower can be divided into 2 parts by only from one plane. If this is your pea flower, you can only divide it into 2 parts from by this single plane, not by any other plane. That is zygomorphic, and this is true. But why is it zygomorphic? Because the estivation is papillonaceous. Papillonaceous means it's talking about vexillary estivation. Okay, in Fabaceae flowers we have vexillary estivation. What is vexillary estivation? There is a standard petal, then there are lateral wings, and then there are anterior keels. Is this shape similar? Little bit, right? So if you divide this flower, you can only divide it into two parts by more than one plane. So due to vexillary estivation, you can say that the pea flower or the Fabaceae family flowers are zygomorphic. Okay? So the answer is 1. Both A and R is correct, and R is the correct explanation of A. Next. Now, there are different enzymes given in list 1. You have to match with their function in list 2. The answer for this is option number 2. So, lipases, what it does, lipases is used in detergent formulation. Pectinases, it will help in clarifying bottled juice. Streptokinase is a clot buster. It busts the clot present inside the body. And cyclosporin is a immunosuppressive agent. Okay, second is the answer. Next, which of the following statements about the cell wall is incorrect? Incorrect. First is viruses are nucleoproteinaceous infectious agent. Yes, viruses are infectious agent. They are made up of nucleoprotein, means nucleic acid and proteinaceous part both. So their genetic material is nucleic acid, and their capsid or the protein part is the capsid, the envelope is your protein. So both nucleic acid and protein are parts of viruses. Second option, the secondary cell wall is formed in the outer side of the primary cell wall in the mature plant cells. It's wrong. If this is your primary cell wall, okay, the secondary, and this is your plasma membrane, okay, this is the cell membrane, and this is the primary cell wall. The secondary cell wall is formed in the inside of the primary cell wall near the plasma membrane. Okay, it's not on the outside. Okay, it's not on the outside, it's inside. Okay, so secondary wall is formed on the outer side of plasma membrane, no, on the inner side of plasma membrane. Okay, second is your incorrect option. Okay, now this question, cell envelope of prokaryotes include glycocalyx, cell wall, and plasma membrane, 3 layers. Okay, next question. For this question, the answer is 4th. Okay, so saprophyte, what are saprophytes? Saprophytes are those who feed on dead and decaying matter. So decomposition of dead organic matter is done by saprophytes. Parasites are those who depend on living plants and animals. Lichens is the symbiotic association between algae and fungi, whereas mycorrhiza is a symbiotic association between fungus and roots of higher plants. So 4th is your correct answer. Okay. Now next question, which of the following kingdoms, this is the last question, okay, for the botany part, which of the following kingdoms show organ level of body organization? Which is the one which shows? The answer is 2nd. Plant and animal, C and D. Monera, Protista, fungi, they don't have any organs. They are very lower level organisms. Okay, only plant and animals, they have organ level body organization. So the answer is 2. So with this, we have completed all the questions for the botany part in your grand test 2. I hope all your doubts are clear. We'll thank you for seeing this, and thank you for attempting the test. Okay. Well, this is it from my side. So everyone take care. Bye. See you. Okay.
Hello everyone. I am Dikshita ma'am, and today we'll be discussing the paper of NEET 2026 Grand Test Series. We'll be discussing the zoology portion of this. It was the level of difficulty in this paper was moderate, and I hope you all did extremely well in this paper. Let us discuss the questions here. First, statement 1 we have habitat fragmentation and genetic drift may accentuate variations leading to appearance of new species and hence evolution. So how does evolution take place? Evolution takes place because of habitat fragmentation and genetic drift, right? Because of these two factors, what is happening? It is leading to the formation of new species, and that will ultimately lead to evolution. So yes, this is a correct statement. Statement 2, the story of evolution of modern man is most interesting and appears to parallel evolution of human brain and language. So as the modern man was evolving, along with it, the brain was also developing, and as a result of a developed brain, what was evolving? Language was also evolving. So this also is a correct statement here. So yes, both the statements here are correct. Option 4 is your correct answer for this question. Right, let's move on to the next question. Xylophyton is not the ancestor of. There is a very beautiful chart given in the evolution chapter where you see how the ancestors of different kinds of organisms actually were, right? So in that chart you will see that Xylophyton was the ancestor of nattales, ginkgos, sphenopsids, but lycopods. Lycopods were not originating from Xylophyton, so Xylophyton is not the ancestor of lycopods. So for this question, your correct answer be number 3. Option 3 will be your correct answer here. Moving on. This is a match the following from the chapter Animal Kingdom. Let us see what is given here. Radula. What is radula? Radula is a feeding organ or a rasping organ. This radula is present in the phylum Mollusca. Phylum Mollusca, from here, which one will be? This is Pila, right? A2. Now we have two options for A2. Next, let us see jointed legs. Jointed legs are always found in one phylum, that is Arthropoda. Arthros, joints, poda, legs or jointed appendages. So jointed legs will be in Arthropoda. Pheretima is not an arthropod. Antedon is not an arthropod. Araneae is actually spiders. So this belongs to Araneae, has jointed legs. Third, we have water vascular system. Water vascular system is a characteristic of the phylum Echinodermata. So let us see which one of them is an echinoderm. Water vascular system is seen in Antedon, A3. Last we see closed type of circulation. In Animal Kingdom, for the first time, closed circulation is seen in the phylum Annelida, correct? And Pheretima or your earthworm is an annelid. So D1. So A2, B4, C3 and D1. Option 2 will be your correct answer here. Option 2 is the correct answer here. Fertilization is internal in. Fertilization is internal in. Hydra, no, it is not in Hydra. Hyla, incorrect. This option is incorrect. Hagfish and catfish, incorrect. Spongilla and nereis, incorrect. Correct option will be filarial worm and tapeworm. That is, in this phylum, we only see internal fertilization, that is, the fusion of the male and the female gamete happens inside the body, not outside the body. Okay, for that reason, option 3 will be the correct answer here. Moving on, study the following statements about frogs. Which of the above are incorrect? Incorrect. Corpora quadrigemina forms the midbrain. This is incorrect. Corpora quadrigemina forms the midbrain of mammals, not on frogs. Incorrect. Receptors of touch, smell and taste are the cellular aggregates around the nerve endings. Correct. The ear is an organ of hearing as well as balancing. Correct. Eyelids are absent. Incorrect. In frogs, there are nictitating membrane that is present. So, eyelids are not completely absent as your NCERT says. For that reason, A and D are your incorrect options. For that reason, option 1 will be your correct answer here. Option 1. Moving on. Okay. The DNA precipitated by chilled ethanol is removed by a technique called. The DNA that is precipitating with the help of chilled ethanol is removed by a technique which we do not call elution, which we do not call PCR, which we do not call agarose gel electrophoresis, but which we call as spooling. Option 3 is your correct answer here, right? Option 3. Okay, moving on to the next question. Study the following statements. Which of the above are correct? We need to identify the correct statements here. Interstitial fluid has the same mineral distribution as that in plasma. This is correct. Lymph is an important carrier for nutrients, hormones, etc. This is also correct. Lymphatic system collects interstitial fluid and drains it back into the major arteries. Incorrect. It is not draining it back to the major arteries but major veins. Fats are absorbed through lymph in the lacteals present in the intestinal villi. Correct. Direct statement from NCERT. So your correct statements are A, B and D. Option 3 will be the correct answer here. Moving on. Okay. Assertion. One assertion reason question let us solve. Assertion. Secretion of most hormones is regulated by negative feedback mechanism. Yes, this is correct. Reason. Secretion of PTH is mainly controlled by anterior pituitary hormones. No. Parathormone is released by parathyroid gland that is not under the control of pituitary gland. So for that reason this is incorrect. For that reason assertion is true but reason is false and option 2 is your correct answer here. Right? Option 2. Okay. In insulin, disulfide bonds are present between. Very easy chain A and chain B because the mature insulin will not have the chain C. For that reason, we will consider option 2 is the correct answer here. Okay. Direct method of gene transfer used to introduce rDNA into animal cells is. The key word here is animal cell and direct method. What we will consider? Microinjection. Microinjection is the direct method by which we transfer the gene into the animal cells directly. And for that reason, your correct answer here will be option 1. Right? Okay, moving on to the next question. Okay, select the incorrect statement. Select the incorrect statement. Decreased glomerular blood pressure leads to release of renin. When the glomerular blood pressure is decreasing, then it will lead to the release of renin. This is correct. GFR is the amount of blood filtered by kidneys per minute. Incorrect. GFR, glomerular filtration rate, is the amount of blood filtered by a nephron or filtered by the glomerulus per minute. Aldosterone leads to increased GFR. This is correct. Renal fluid gets diluted in ascending limb of loop of Henle. Again correct, because in the ascending limb of loop of Henle is permeable to electrolytes. It is permeable to salt. For that reason the fluid becomes diluted. Your correct answer here is option 2. Why? Because it is incorrect. Right? So moving on to the next question. Statement 1. During blood clotting, an enzyme called thrombokinase converts thrombin into prothrombin. Is this correct? This is incorrect, because during blood clotting an enzyme complex called thrombokinase will convert prothrombin into thrombin. So for that reason incorrect. Thrombokinase is formed by a series of linked enzymatic reactions, which is known as a cascade process, involving a number of factors present in plasma in an inactive state. Yes. For that reason statement 1 is incorrect, but statement 2 is correct, and for that reason option 2 is your correct answer here. Okay, moving on. Okay, study the following regarding the transport of gases in humans. Which of them are correct? Correct statements we need to find. Effect of concentration of carbon dioxide and hydrogen ions on the affinity of hemoglobin with oxygen is called chloride shift. This is not chloride shift. This is not chloride shift. This is incorrect. Most of carbon dioxide is transported from tissues to lungs as bicarbonates. Yes, this is correct, because the amount of carbon dioxide being transported is the highest through bicarbonates, that is 70%. Correct. In blood, most of the oxygen is transported by plasma. Incorrect. In blood, most of the oxygen is being transported by RBC with hemoglobin, not in plasma. D. Entry of bicarbonates into RBC and exit of chloride ions from RBC takes place at tissues. Again, incorrect. So your correct answer here is only B, and for that reason option 4 is your correct answer here. Option 4. Right, moving on. Okay, let us see and match the following from the chapter Human Reproduction. Oviducts, that is fallopian tubes. Oviducts have simple ciliated columnar epithelium. Why? So that the transport of the ovum, egg, happens smoothly from the ovary to the fallopian tube for fertilization. B. Ducts of glands will have simple cuboidal epithelium, and walls of blood vessels have endothelium. And what is endothelium? Simple squamous epithelium. And lastly, lining of pharynx. Lining of pharynx will have non-keratinized stratified squamous epithelium. So A3, B2, C5 and D4. Option 2 will be your correct answer here. Option 2 will be the correct answer here. Okay, moving on to the next question. Which of the following are autoimmune disorders? Autoimmune disorders. When our body is harming our own body, that is when the body cannot distinguish between self and non-self cells. Okay. Myasthenia gravis is an autoimmune disease. Correct. Rheumatoid arthritis, yes. Gout is not an autoimmune disease. Gout happens because of accumulation of uric acid. It has nothing to do with autoimmune disorder. Muscular dystrophy is a genetic disorder. Incorrect. Systemic lupus erythematosus, yes, this is an autoimmune disorder. So A, B and E. Option 2 will be the correct answer here. Option 2 will be the correct answer here. Moving on. In humans, lymphocytes interact with antigen, then proliferate to become effector cells, where they are becoming in the effector cells, not in the thymus, not in the red bone marrow, not yellow bone marrow, but in the lymph nodes. In the lymph nodes, the lymphocytes interact with antigen, then proliferate to become effector cells. Option 2. Option 2 will be the correct answer here. Moving on. Peptide hormone that binds with membrane receptors is peptide hormone. Be very careful in such type of questions. Peptide hormone. Will it be adrenaline? No. Catecholamine. Will it be aldosterone? No. Membrane receptor. Oxytocin. Yes, because they are asking for membrane receptor. Oxytocin is a peptide hormone and has a membrane receptor, not intracellular receptors, right? For that reason, option 3 will be your correct answer here. Option 3. Moving on. Statement 1. Peptide bond is present in all 4 levels of protein structure. Yes. Peptide bonds which is present between one amino acid and another amino acid, for example, have to be present in all 4 levels of protein structure, that is primary, secondary, tertiary, quaternary. Otherwise, how will a protein form? Otherwise, how will the amino acids bind with each other and form the protein, right? For that reason, this is correct. Statement 2. Every enzyme to show its catalytic activity requires a cofactor that is non-proteinaceous but organic in nature. Not necessarily. Not necessarily. For that reason, incorrect. Statement 1 is correct, but statement 2 is incorrect. Option 3 will be your correct answer here. Okay. Which of the following is not true about thyroxine? They are asking not true. It maintains water and electrolyte balance. Yes, that is done by thyroxine. It supports the process of RBC formation. Yes, that is done by thyroxine. It regulates metabolism of proteins. Yes. It decreases body temperature. No. This function is not done by thyroxine, and for that reason, this will be your correct answer here. For that reason, option 4 will be the correct answer here. Okay, moving on. Okay. Study the following statements. Which of the above are correct? Ejaculatory ducts lead into prostatic urethra. This is correct. Bulbourethral glands are located below the prostate gland on either side of membranous urethra. This is also correct. The testis is covered by a dense covering called tunica albuginea. This is also correct. Are all the statements correct here? Let us find out. Each seminiferous tubule is lined on its inside by male germ cells. Yes. And Sertoli cells, not Leydig cells. For that reason, this is incorrect. We are asked to select the correct statement, so we will select A, B and C, for which option 3 will be your correct answer here. Option 3. Option 3. Moving on. Okay. Next question, number 38. Identify the incorrect statements and choose the appropriate option. Incorrect. Okay. A. DCT is capable of selective secretion of hydrogen ions, potassium ions and ammonia to maintain pH and ionic balance in blood. This is correct. Nitrogenous wastes are reabsorbed by active transport. Incorrect. Incorrect. They are not reabsorbed by active transport. Incorrect. C. Conditional reabsorption of sodium and water takes place in DCT. This is also correct. In DCT, in distal convoluted tubule, whatever reabsorption is happening, it is only conditional and what absorption is happening? Of sodium ions and water. So this is correct. Next, DCT reabsorbs more glucose compared to PCT. Impossible. PCT will always reabsorb the highest amount of nutrients. No other part of the nephron can reabsorb nutrients as much as PCT does. Highest amount of reabsorption will happen in PCT. For that reason, this is incorrect. E. Reabsorption of water occurs passively in the initial segments of nephron. Yes, this is correct. So, our incorrect answers they have asked, it will be B and D. It will be option 1. This will be option 1 will be the correct answer here. Moving on. Study the following statements about humans. Which of the above are incorrect? Which of the above are incorrect? The heart is a mesodermally derived organ situated in the thoracic cavity. Correct. Heart is derived from mesoderm, that is the mesodermal layer, which is the germ layer, and it is present in our thoracic cavity. Correct. B. Kidneys are situated between the levels of third lumbar vertebra and last sacral vertebra. Incorrect. This is not correct. Brain is covered by cranial meninges consisting of an outer dura mater, a middle arachnoid and an inner pia mater. So this is also incorrect. Incorrect. Last. The dorsal ends of 8th, 9th and 10th pairs of ribs do not articulate directly with the sternum but join the 7th rib with the help of hyaline cartilage. So, is this correct? This is almost correct, right? But what are they saying? They are saying dorsal ends. The dorsal end is not connecting to our sternum. The dorsal end is connecting with the vertebral column, right? So, for that reason, this is also incorrect. So for that reason, the incorrect options will be B, C and D. Option 3 will be your correct answer here. Option 3. Okay. Moving on, option 3. In blue-white screening method, the rDNA containing cells on nutrient medium give dash color colonies. If a cell will have, it's a rDNA containing cell, if the cell will have rDNA, then it will not be chromogenic, which means it will not impart color, which means it will be white in color. For that reason, option 3 will be your correct answer here, right? From blue-white screening. If it has rDNA, white colonies. If no rDNA, then blue colonies. But since our question had rDNA, so our answer will be white colonies. So option 3 will be your correct answer here. Moving on. Match the following joints with the bones. Match the following joints with the bones. Okay, let us see. Gliding joints. Gliding joint is present in between the carpals. Hinge joint is present between the humerus and the ulna. B4. C. Pivot joint is present between atlas and axis, that is on here at the base of our head. And lastly, saddle joint, that is present between the carpal and metacarpal of our thumb here, right? So, A3, B4, C2 and D1. Yes, option 2. Option 2 will be the correct answer here, 2. Okay. One more statement based question. Statement 1. LH stimulates secretion of androgens in males, whereas in females it promotes ovulation, formation and maintenance of corpus luteum. Is this correct? Is this correct? Yes, this is correct. In males, what will LH do? That is luteinizing hormone will do. In males, it will help in the secretion of androgen, right? But in females, LH will help in the bursting of the Graafian follicle to release the ovum, which is ovulation, and because of the Graafian follicle bursting, the Graafian follicle, the burst Graafian follicle becomes the corpus luteum, and that formation and maintenance is also done by the luteinizing hormone. So this is correct. Second, FSH and androgens regulate spermatogenesis. Correct. Follicle stimulating hormone and androgens regulate spermatogenesis. Both the statements 1 and 2 are correct. Option 4 will be the correct answer here. 4. Okay. Next, question number 45. The ovarian hormone that acts on the mammary glands and stimulates the formation of alveoli and milk secretion is. Okay, very easy. Why very easy? Because what they have mentioned in the question, they have mentioned ovarian hormones. Which are the ovarian hormones? Estrogen and progesterone. What do we have here? Progesterone, right? So we will not even look at the other options because ovarian hormones are just estrogen and progesterone. So the answer will be progesterone. But still, let us see. Prolactin is not released by the ovary. It is released by the pituitary gland. Oxytocin also pituitary gland. Somatocrinin also not released from the ovary, right? So for that reason, option 2 will be your correct answer here. Moving on. Which of the following statement correctly describes the action of acetylcholine at a chemical synapse? 1. It opens the ion channels in the postsynaptic membrane. Will it happen? Of course it will happen. Why? Because what is happening? Acetylcholine is being released, right, in the synapse. So this acetylcholine will go and bind to the receptors which are present in the postsynaptic membrane. Because of this acetylcholine binding with the receptors of the postsynaptic membrane, this will lead to the change in polarization, and for that reason the ion channels will open in the postsynaptic membrane. So for that reason, option 1 is your correct answer here, but let us see why the other options are incorrect. It always increases the potassium concentration inside the axon. Not necessarily, not necessarily. It maintains the resting potential. No. It always increases the sodium concentration outside the axon. Again, not necessarily. For that reason, option 1 will be your correct answer here. Moving on. When you hold your breath, when we are holding our breath, which of the following gas changes in blood would first lead to the urge to breathe? That is, falling oxygen concentration in blood, rising carbon dioxide concentration in blood, falling carbon dioxide concentration in blood, or rising oxygen and falling carbon dioxide concentration in blood. Rising carbon dioxide concentration in blood, because when we are holding our breath, it means that the carbon dioxide that is produced inside the body do not have a outlet to go, right? For that reason, the carbon dioxide is building up in the body, and for that reason, that will be the first urge to breathe. That will lead to the first urge to breathe, and for that reason, option 2 is your correct answer here. Option 2. Okay. Which mouth parts are present on the lateral side of the pre-oral cavity in cockroach? Labrum. Labrum is also known as the upper lip. So this is present anterior, not lateral. They have asked for lateral. Mandibles, yes. Mandibles, yes. Labium, not labium. Labium is in the posterior part. So mandibles, yes. Maxillae, yes. So for that reason, mandibles and maxillae will be the correct answer here. Hypopharynx is not present laterally. And yes, option 3 is your correct answer here. Option 3. Moving on to the next question. Starch forms blue color with iodine because. Why is starch forming blue color with iodine? it is a storage and branched polysaccharide? No. No. It contains complex helices and hence can hold iodine? Yes. It has many helices, and in that helices what is present? Iodine can be stored, and for that reason it gives the blue color when it reacts with iodine. Third, starch breaks down when it reacts with iodine? Incorrect. It is a disaccharide. No, it is not a disaccharide. So for that reason option 2 is your correct answer here. Moving on. Next question. The correct statements about the working of oral contraceptive pills are: oral contraceptive pills inhibit ovulation and implantation? Correct. B. Alter the quality of cervical mucus to prevent or retard the entry of sperms? Correct. Inhibit the formation of primary follicles? No. It cannot inhibit the formation of primary follicles. It will be formed regardless. For that reason, A and B are correct. We need to choose the correct answer. So for that reason, option 2 is your correct answer here. Option 2. Moving on to the next question. Which of the following destroy our infected body cells in innate immunity? Innate immunity, okay? Antibodies released by memory cells. This is not a part of innate immunity. This is a part of adaptive immunity. Antibody or cell mediated immunity. Antibodies released by plasma cells. This is also not a part of innate immunity. Both these are part of not a part of innate immunity. So incorrect. 3. Cytotoxins released by TC cells. Again, this is also TC cells. Cytotoxic T cells are not a part of innate immunity. Incorrect. Cytotoxins released by natural killer cells, cellular barriers, right? For that reason, option 4 will be your correct answer here. Why are the other options incorrect? Because they are not a part of the innate immunity. Because what is the question asking? Which of the following destroy our infected body cells in innate immunity? Right? These are not part of innate immunity. For that reason, option 4. Select the vector-borne helminth disease from the following. Vector-borne helminth disease, which means worms. Typhoid? No. Bacterial? Malaria? No. The vector here is the mosquito, arthropod, not worms. Common cold? Virus? Incorrect. Filariasis? Filarial worm. For that reason, option 4 is your correct answer here. Option 4. Okay, one more. Match the following. Let us see. Okay, this table is from the chapter biomolecules. If you see, there is a beautiful table of protein and different types of protein and what are its functions. This question is from that table. Let us see. Collagen. Collagen is an intercellular ground substance. GLUT4. GLUT4 is a membrane protein. Inulin. Inulin is a polysaccharide. But inulin is not a protein. Inulin is a polysaccharide, so it is a carbohydrate. Antibody. Antibody is fighting infectious agents. So from here, most of them are from that table except for inulin, which is a polysaccharide. So correct answer here is A4, B2, C1, D3. Option 2 is your correct answer here. Option 2. Okay, next, one more assertion and reason question let us solve. Assertion: when a neuron is suitably stimulated, an electrical disturbance is generated. Yes, when a neuron is suitably stimulated, an electrical disturbance is generated because of the change in polarization. For that reason, neuroglia make up more than 1/2 the volume of neural tissue in our body. Yes, our nervous tissue is made up of two types of cells. One is neuron and the other is neuroglial cells. neuroglial cells are present more than 50% of the, is making up more than 50% of the nervous tissue in the body. This is correct. But this is not explaining assertion, right? These are two very separate statements. Reason is not explaining assertion. For that reason, option 2 will be your correct answer. Both assertion and reason are true, but reason is not the correct explanation of assertion. Option 2. Next, let us see one more statement based question. Statement 1. Severe anemia and jaundice are the clinical manifestations of erythroblastosis fetalis, which is correct. Statement 2. Rh antigens of the Rh positive fetus do not get exposed to the Rh negative blood of the mother in the first pregnancy during gestation, as the two bloods are well separated by the placenta. This is also correct. So both statement 1 and statement 2 are correct here. Option 1 will be the correct answer here. Option 1. Okay. One more sequence type question let us solve. What is the correct sequence of occurrence of the following reproductive events? So among all these events, the first event that will occur will be insemination. This is the first one. That is the transfer of the male gametes into the female reproductive tract. After insemination, what will happen? Capacitation of sperm. Capacitation of sperm will happen, which will, once the sperm has reached a female reproductive tract, it starts capacitation reaction. Next what will happen? The sperm will reach the fallopian tube of the female reproductive tract, and because of this, the sperm will now fuse with the ovum, which will lead to fertilization, and this is the third step. After this, what will happen? After the fusion of the male and the female gamete, it will result in a structure which is known as zygote. This zygote is a unicellular structure, but the zygote will start dividing 2 cells, 4 cells, 8 cells, right? And this will lead to a structure called blastocyst, which is blastulation. D. After that, there will be implantation, which will happen in the uterus. That is option E. And after that, and after implantation, the structure will still start growing, the cells will start dividing, which will lead to gastrulation. So for that reason, your correct sequence will be B, A, F, D, E and C. For that reason, option 4 will be the correct answer here. Option 4. One more question, let us solve the next question. Assertion. Again, assertion reason. Oral contraceptive pills which contain progestogen estrogen combination inhibit ovulation. Correct. As we have discussed in one of the previous questions also. Correct. Oral contraceptive pills which contain progestogen estrogen combination will prevent ovulation. Correct. Reason. Such oral contraceptive pills inhibit secretion of gonadotrophins. This is also correct. Because such oral contraceptive pills is preventing secretion of gonadotrophins. What are the gonadotrophins? LH and FSH. For that reason, what will happen? Ovulation will not happen, and that is why it is inhibiting ovulation. So yes, both your assertion and reason are correct, and reason is correctly explaining your assertion. So option 4. Option 4 will be your correct answer. Both of them are correct and reason is correctly explaining assertion. Option 4. Moving on to the next question. Okay, one more question from cockroach. Let us see. Which of the above are found in the prothorax of the cockroach? Now, if you know, if you remember, the entire body of the cockroach is divided into head, thorax and abdomen. The thorax is divided into 3 parts: prothorax, mesothorax, metathorax. Now the question is, what is not found in prothorax? Let us see. Nerve ring. Oh sorry, what is found? What is found in the prothorax? Nerve ring. Nerve ring is not found in prothorax, it is present in the head. Spiracles are not found in prothorax. Legs, yes. Each part, each segment of the thorax, that is prothorax, mesothorax, metathorax, will have a pair of walking legs. So yes, legs are present in prothorax. Segmental ganglia, yes, segmental ganglia will also be present in the prothorax. Wings, wings are not found in prothorax because in cockroach it only has 2 pairs of wings. So one pair from mesothorax and the next pair from metathorax. Prothorax will only bear walking legs, not wings, right? For that reason, C and D, option 1 will be your correct answer here, C and D. Select a common feature in labio and lamprey. Labio is an osteichthyes, a bony fish, and lamprey is a cyclostome. What is a common feature between them? Absence of paired fins, incorrect, they do not share this characteristic. Ventral heart, yes, since both of them are vertebrates, what will happen? They will, or both of them are chordates, they will have a ventral heart that is present in the front. So for that reason, this is a common feature in labio and lamprey. Absence of scales, it is absence of scales is only seen in lamprey, not in labio. Presence of jaws is only seen in labio, not in lamprey, because they are jawless fishes. For that reason, option 2 will be your correct answer here, right? Option 2. Moving on, let us see what this says. Main steps in the formation of recombinant DNA are given below. Arrange these steps in a correct sequence. Let us arrange these steps in a correct sequence. First of all, we will cut the DNA, right? We will cut the DNA with the help of molecular scissors or restriction enzymes. So cutting of DNA at specific location by restriction enzyme will be done. After that, what we will see is, we will isolate the desired DNA fragment, right? This will be the second step. The third step is that whatever we have isolated, we will amplify that part. So amplification of gene of interest using PCR, this will be the third step. And lastly, insertion of recombinant DNA into the host cell. This will be the fourth step. So B, C, D, A. Option 4 will be your correct answer. That will be the proper sequence where how our DNA technology is happening. Right, moving on to the next question. One more statement based question. Let us see what it is saying. Statement 1. Fitness is the end result of the ability to adapt and get selected by nature. Fitness is the end result of the ability to adapt and get selected by nature. Yes, correct. Yes. 2. Fitness is based on characteristics which are inherited. Yes, this is also correct, right? So both the statement 1 and 2 are correct here, and option 4 will be your correct answer here. Option 4. And with that, we come to an end of the zoology portion of this paper. I hope you all did amazing, and we shall meet in the next paper to discuss. Okay. All the best, guys. Bye. Keep learning.
Hello students, welcome to the session. Today we are going to discuss about NEET Grand Test Series Paper 2. So let us start. The question paper is very easy and moderate and well balanced. You are going to get the same type of questions in the NEET 2026. So let's start without any delay. A question is asking, in 1 liter flask, 1 mole of N2O4 is taken and allowed to attain equilibrium at a high temperature. According to the reaction, N2O4 gives rise to NO2. At equilibrium, the degree of dissociation is given as 0.5, and calculate the value of the equilibrium constant. So how to calculate the equilibrium constant? You can say Kc is equal to here NO2 square and N2O4. So according to this equilibrium N2O4, you can say, and it is reversibly given rise to 2 NO2. Initially it is taken as 1 mole per 1 liter and equilibrium is not reached. Once the equilibrium reached, degree of dissociation is alpha is going to be 0.5, 1 minus 0.5, and it is going to be 2 into 0.5 is going to be 1 is going to be 0.5. So you can say here it is going to be 1 square upon 0.5. You are going to get answer as 2. So equilibrium constant Kc, answer is going to be 2 for this question. Option 2 is the correct answer for this question. Students, this is going to be the thermodynamics question, and it is asked for ideal gas in isothermal process, which of the following graph is correct? It is clearly said that isothermal process. In the isothermal process, internal energy is going to be 0. There is no change in internal energy. If we talk about internal energy with the change in the volume, internal energy kept constant. So option 1 is the correct answer for this question. Students, this question based on the thermochemical equations and enthalpy of neutralization. So it is given as heat of neutralization of a strong acid and a strong base is going to be minus 57 kilojoules. It is given 200 centimeter cube milliliters of 0.1 molar H2SO4, N factor is going to be 2. So number of equivalents of H2SO4 is going to be 200 into 2 into 0.1 is equal to 40 milli equivalents. And it is mixed with the potassium hydroxide, its N factor is going to be 1. So milli equivalents of potassium hydroxide is going to be 150 into 0.2 is going to be 30 milli equivalents. So that you are going to neutralize these 30 milli equivalents of H2SO4 only. So for 1000 milli equivalents, you are going to release minus 57.1, 57 kilojoules. If it is for 30 milli equivalents, how much? You are going to get 57 into 30 upon 1000. You are going to get 1.7 kilojoules. So the answer for this question is going to be 1.7 kilojoules is going to be the answer. Option 1 is the answer for this question. This is the question from chemical bonding dipole moment. The dipole moment of this halobenzene is 1.5 dBi, and the dipole moment of this trihalobenzene is given. When you see the bond moment is going to be cancelled here, as a result of the resultant dipole moment becomes 0. Therefore, option 3, 0 is the correct answer for this question. Students, this is the structure based on Buckminster-Fuller C60. Number of 6-membered rings and 5-membered rings in a fullerene is X and Y. Number of 6-membered rings are 20. Number of 5-membered rings are 12. So X is equal to 20 and Y is equal to 12. And the difference between X minus Y equal to, so X minus Y is equal to 20 minus 12 is equal to 8 is coming. Now the question is asking about that equal to atomic number of carbon, it is 12, so it cannot be. Total number of bonds in XeO4. If you see the XeO4 structure, Xe double bond O, double bond O, double bond O and a double bond O. So there are 8 bonds and this 8 is matching here. Option 2 is correct. Oxidation state of sulfur in SO3 is +6, which is not correct. and maximum covalency of oxygen is 3, which is not correct. So for this question, option 2 is going to be the correct answer for this question. Students, this question is based on halogens group 17 elements. Halogens are the most reactive elements in the periodic table. Regarding the properties of halogens, improper match is. Let us see various things. It is given F2Cl2Br2I2 oxidation power decreases, so it is a correct one. I2Br2Cl2F2 melting points, so melting points increases down the group, it is also correct. And chlorine, fluorine, bromine, iodine, because of a small size, electron affinity, electron gain enthalpy of fluorine is less than chlorine. So chlorine is greater than fluorine, which is a well-known fact. This is also correct. Now you can see here, fluorine, chlorine, bromine, iodine bond dissociation energy due to small size fluorine and fluorine, the lone pair of electrons on the fluorine experiences inter-electronic repulsions. That is the reason why fluorine has the very low bond dissociation energy. It is given highest bond dissociation energy, which is actually not correct. So that this is an incorrect match, but it is incorrect match is asked as a question. So option 1 is the correct answer for this question. Students, this question is based on molecular orbital theory. So given statements, bond order of H2 plus is equal to H2 minus. Let us say H2 plus, it is having just one electron. You are going to get sigma 1s1, and H2 minus, you have 3 electrons. So you have sigma 1s2, sigma star 1s1. Now you can see the bond order is going to be, so 1 minus 0 by 2 is equal to 0.5. That is going to be 2 minus 1 by 2 is equal to 0.5. He wanted to say bond order is correct. Yes, it is correct. Stability of H plus is equal to H minus. Absolutely not. Why? Because it has one electron in anti-bonding molecular orbital. So I don't accept this. So statement 1 is correct. Statement 1 is true and statement 2 is not correct. So the reason is presence of electron in anti-bonding molecular orbital makes it less stable on it. So option 3 is the correct answer for this question. Here it is one more question from the chemical bonding. The bond angle in H2O is 104.5 degrees, is higher than the bond angle of H2S 92.1 degrees. The difference is mainly due to difference in the number of valence electrons. Absolutely not correct. Both have the same number of valence electrons because they belong to the same family. Difference in the electronegativity of oxygen and sulfur. So this is the answer for this question. So because of the electronegativity, the bond angle varies here. So electronegativity of the central atom decreases, bond angle decreases in this case. Children, here it is a one more question from the biomolecules. So which is not a disaccharide? Maltose is a disaccharide between two glucose. It's a disaccharide. Galactose is a monosaccharide. Lactose is a disaccharide of glucose and galactose, and sucrose is a disaccharide of a glucose and a fructose. So here among 4, option 2 is the only not a disaccharide. So option 2 is the correct answer for this question. Students, here it is one more question. Given below are 2 statements. Assertion: acid catalyzed hydrolysis of ethyl acetate is a second order reversible reaction. Acid catalyzed ester hydrolysis, that what we study, is a popular example of pseudo first order reaction. And order of reaction is always depends on stoichiometry of the reaction, which is absolutely not correct. Order of a reaction depends on rate law, which is experimentally determined. It is not at all related to stoichiometric coefficients. So I want to say option both A and R are false. So it is going to be option 4 is the correct answer. Here it is one more important question, logical question. NTA is always asked about this type of question. A dry cell with an EMF 1.5 volt is vertically cut into two halves. Each piece as obtained is made into a dry cell, and thus starting functioning again the EMF of the newly obtained cell in volts. So if you take a battery like this, of course if you cut into half, it will not work because of the mechanical damage. Let us hypothetically assume we have taken the content of the electrode and electrolyte is into half. It will not impact the voltage of this galvanic cell. The voltage of the galvanic cell is dependent on the concentration, not the quantity. It is not dependent on the size of the electrode as well. So whatever the voltage here mentioned here, the same will be there. Such a nice logical question. I love it. Option 3 is going to be the correct answer for this question. Students, this is question based on direct application of Nernst equation. So the cell potential for the given cell at 298 Kelvin, platinum, hydrogen 1 bar, H plus aqueous, copper 2 plus aqueous and copper solid. And the EMF is given 0.31 volts. This is EMF, and the pH is given will be 3, and you are asked to calculate copper 2 plus concentration. So let us apply directly. EMF of the cell is equal to E naught minus 0.0591, you can write 0.06 by n factor, and a log H plus square and a copper 2 plus. So let us equate all these things here. So EMF is given 0.31 and E naught cell is given as 0.34 minus 0.06 upon n factor is going to be 2 and log H plus ion concentration is given. pH is equal to negative log H plus ion concentration, and H plus ion concentration is going to be 10 to the power of negative pH. That is going to be 10 power minus 3. pH is given 3. So I want to say 10 raised negative 3 square. You have to calculate copper 2 plus concentration. So on doing so, copper 2 plus concentration comes to be 10 raised negative 7. So I want to say is 10 raised negative x is going to be 7. So 7 is the answer. Option 3 is the correct answer for this question. Students, this is the percentage of isotopic abundance based question. The isotopic abundance of C12 and C14 is 98% and 2% respectively. What would be the number of atoms in C14 isotope nearly in 12 gram of carbon sample? If it is for C14, okay, so it is going to be 6 into 10 raised 23. So if it is a 14, if it is a 12, it is going to be how much? For 100%, it is said that 12 into 6 into 10 raised 23 upon 14. If it is for 98% and 2%, for 100% 12 into 6 into 10 raised 23 by 14. For the 2% of the C14, you are going to get 2 into 12 into 6 into 10 raised 23 by 14. On solving, you are going to get 1.2 into 10 raised 22 is the answer. Option 1 is the correct answer for this question. Here is the question, my dear students. 10 grams of sodium hydroxide is dissolved in 10 ml of 5 molar HCl solution. Excess of HCl is neutralized by 5 ml of 2 molar barium hydroxide solution. So the percentage of purity of this caustic soda NaOH is going to be. So let us talk about number of equivalents of HCl. So number of equivalents of HCl, it is going to be 10 ml of 5 molar. So it is going to be 50 equivalents. Equivalents of barium hydroxide, that is given as 5 ml and 2 molar, 5 ml and 2 molar, and its N factor is going to be 2. So it is going to be 20 equivalents. So when this is neutralized, 50 milliequivalents and 20 milliequivalents, so number of moles of HCl left, number of equivalents of HCl left is equal to 50 minus 20, that is equal to 30 equivalents, that is neutralized by NaOH. So number of equivalents of sodium hydroxide is equal to 30 equivalents, and N factor is 1, and the mass of the sodium hydroxide is going to be, weight of sodium hydroxide is going to be 30 into 40 is going to be 120 milligrams, because these are all taken into milli equivalents, right? These are all taken into the milli equivalents. So it is going to be 1.2 grams. So 1.2 grams means it is going to be 12% is the answer. The percentage of purity is going to be 12%. Option 2 is the correct answer for this question. Students, this is a question based on the buffers. Ka of acetic acid is 2 into 10 raised negative 5. Buffer action of CH3COOH and CH3COONA is found to be maximum at the pH. Let us talk about Henderson-Hasselbalch equation. It is going to be pH is equal to pKa plus log salt by acid concentration. Buffer capacity is going to be maximum whenever salt concentration is equal to acid concentration. So if that is the case, pH is equal to pKa is equal to, Ka value is given, negative logarithm to the base 10 of Ka value, that is going to be negative log 2 into 10 raised negative 5. So 5 minus log 2. So 5 minus 0.3 is equal to 4.7 is the correct answer for this question. Option 2 is the answer for this question. Children, here again one more statement 1 and statement 2 question. The statement 1 is saying pH of 0.1 molar ammonium cyanide is equal to pH of 0.01 molar ammonium cyanide. Now you can see concentration is different here, but he is saying that pH is same because it is correct, because ammonium cyanide is a salt of weak acid plus weak base. Therefore, this statement I can say it is the correct statement. And sodium bicarbonate, you can say NaHCO3 on ionization gives rise Na plus and HCO3 minus. Still it can donate a proton to gives rise CO3 2 minus and H plus. So 2 minus. So it is an acidic salt. So it is an acidic salt. I want to say both statement 1 and statement 2 are the correct. So option 1 is the correct answer for this question. Students, this question is a multi-statement question. Regarding the normality, which of the following is incorrect? Finding is incorrect. Let us say normality is equal to number of milliequivalents per milliliter of solution. Normality according to the definition, number of equivalents per liter of solution. or number of milliequivalents per milliliters of solution. Correct statement. For a same solute, the concentration of one normal solution is always less than 1 molar solution. That you can't say. Normality is equal to molarity into n factor. So, it is based on its n factor. So, I would say this is a wrong statement. Let us say on dilution, a solution normality decreases, but number of equivalents of the solute remains same. So, normality is equal to number of equivalents upon volume. Okay, you want to say if volume is there, if you increase, NV is going to be constant. So, equivalence will not change. So, I want to say this is correct statement. In HNO3 solution, it contains 1 mole or 1 equivalent of HNO3 in 1 liter of solution, 1 normal solution. So, molarity is equivalent to the normality in this case because it is 1 mole, 1 equivalent, both are same because n factor is going to be 1 for this HNO3. So, I want to say this is also correct statement. Among all, option B is going to be, so only B, option 1 is the correct answer for this question. Children, this is a logical question based on the solutions and colligative properties vapor pressure concept. Which statement is correct? Let us observe this. 100 milliliters of ether is taken in a vessel and it is transferred to 200 milliliters at the same temperature. This is the case 1, and it is transferred to 100 milliliter capacity, but the temperature is increased. So, vapor pressure of the solution is directly proportional to the temperature. Okay. So, I mean to say as temperature increased, vapor pressure is going to be increased, but the same temperature vapor pressure is going to be the constant here. So, when you talk about this, vapor pressure remains same in the case 1, and while increasing the temperature, the vapor pressure is going to be increased in the case 2. So, for this question, I would say option 4 is the correct answer for this question. Students, here is a question asking about 7.483 into 10 raise 5 Newton per meter square osmotic pressure is found to be 0.1 molar KXFeCN6, which dissociate 50%. The value of X is. So, it is a multi-concept question. Directly it is related to want half factor. We have pi is equal to I CRT, but it is the units is given in Newton per meter square. 1 ATM is equal to 10 raise 5 pascals. So, I want to say pi is equal to I value I wanted to calculate. So, 7.483 into 10 raise 5, directly I will take in the pascal, and I value, and CE I want to say here concentration. How much concentration is given? 0.1 mole. So, you can say this is into 0.1 mole means 0.1 into 10 raised 3 mole per meter cube. So, directly I can say 0.1 into 10 raised 3, then R value is going to be SI units 8.314 into temperature, it is given as 300 Kelvin. On doing so, I value is going to be 7.483 into 10 raised 5 upon 0.1 into 10 cube into 8.314 into 300. On solving so, I value is going to be nearly comes to be 3. But K4FeCN6 or KXFeCN6, it is dissociated into, in which format? It is a dissociation. Whenever it is dissociated, alpha is equal to I minus 1 by N minus 1. So, if that is the case, 50% is taken. So, it is going to be 0.5, alpha value 0.5, I value just calculated 3 minus 1 and N minus 1. On solving, you are going to get N value as 5. When you have N value as 5, Kx FeCN6 and it is going to be X number of potassium plus and FeCN6 is going to be the total particles. Total particles is coming 5. I mean to say X plus 1 is equal to 5. So, X is equal to 4. So, I want to say the formula of the compound is going to K4 FeCN6. So, X is equal to 4. The answer for this question is going to be option 4 is the correct answer for this question. Students, this question is from structure of atom. The question is asking the ratio of number of revolutions of electron per second in second and third orbit of hydrogen atom. Here it is given as V is the velocity, R is the radius. So, number of revolutions of electron per second is said to be orbital frequency, that you can say frequency is directly proportional to velocity by R. So, I mean to say that is equal to frequency of this second orbit to frequency of this third orbit is equal to V2 by R2 into R3 by V3. So, the answer for this question is going to be option 2 is the correct answer. Students, this question is from again atomic structure. An excited electron from hydrogen atom emits a photon of wavelength lambda and it returns to the ground state and the principal quantum number excited state is given as. So, it is coming to the ground state. So, N1 is equal to 1 and it is gone somewhere, and what is the excited state? N2 you have to identify for hydrogen atom. For a hydrogen atom, 1 by lambda is equal to R into 1 by N1 square minus 1 by N2 square. So, you have to solve this N2 value, and that is given as 1 by lambda. So, 1 by lambda R is equal to 1 by N1 square minus 1 by N2 square. It is coming to be 1, so that it is coming to be 1. I want to say 1 by lambda R is equal to 1 minus 1 by N2 square. You can also say 1 minus 1 by lambda R is equal to 1 by N2 square. On solving, so you are going to get N2 is going to be under root lambda R upon lambda R minus 1. So, I would pick option 4 is the correct answer for this question. Students, this is a zero order kinetics reaction. For a zero order reaction, the initial amount of reactant is 100 grams and its half life is 20. The amount of reactant left after 40 minutes. So, let us solve systematically, and simple trick also I just want to give that. So, T half for zero order is equal to initial concentration by 2K. So, K is equal to initial concentration by 2 T half, that is equal to initial concentration, you got it is 100 and 2 into 20 is equal to 10 by 40, I can say that is the rate constant for this zero order kinetics, and amount that is left. So, as we know, for zero order reaction K is equal to A naught by AT. For zero order integrated expression K is equal to A naught minus AT by T. K value you got it as 10 by 40. Initially you have taken, AT you have to calculate when the time is going to be 40 minutes. You are going to get, on solving, AT is going to be 0. So, you won't leave anything. Answer is going to be 0. Otherwise, in zero order kinetics, you can also use simple trick. If you take 100 grams to make it 50 grams, you take 20 minutes. If you spend 20 more minutes, from here it is going to be 50, so from here it is going to be 0. For it is the kinetics of this 0 order kinetics, and 0 order kinetics can be 0, but for first order kinetics it takes infinite time to 100% completion. So answer for this question is going to be option 1 is the correct answer for this question. Students, here is the question based on the match the list 1 with list 2, choose the correct option. So here are the certain combinations are given and their physical appearances come in. When you say pick the known one, rotten egg smell. So whenever FeS is going to react with H2SO4, you are going to get H2S gas. So it is going to be rotten egg smell. And you see FeH2O 6 times SO4 NO, you are going to get a brown ring. So you are going to get a brown ring. So FeH2O and NO is going to NOH2O 5 times, NO is going to form as a brown ring complex. And ammonium hydroxide plus HCl, it is going to give us ammonium chloride as white fumes. And when NaOH and Cr2 O2Cl2, you are going to get CrO2Cl2, you are going to get lead chromate. So you are going to get chromyl CrO4, NaCrO4 is going to get Na to CrO4, your lead solid, sodium chromate is going to be formed. So here the answer for this question is going to be A2, B3, C1, D4. Option 4 is the answer for this question. Here it is a one more question related to hydrocarbons here. When N-hexane is treated with MO2O3 molybdenum trioxide at 500 degrees centigrade at 10 to 20 atm, the change taking place, you are going to get N-hexane 1, 2, 3, 4, 5, 6. This is N-hexane. When it is heated along with molybdenum trioxide and high pressure, you are going to get benzene. So let us talk about the facts about the benzene. Hybridization of the carbon changes. It was sp3 and it become sp2. And number of carbon to carbon bonds increases and number of carbon to hydrogen bonds decreases and the percentage of carbon increases. So I would say A, B, C, D all are correct regarding this. Answer 1 is the correct answer for this question. So here it is a practical organic chemistry related question. Identify the incorrect statements from the following. Benzaldehyde gives a positive Fehling's test and a Tollens test. Benzaldehyde gives a Tollens test but not a Fehling's test. Benzaldehyde gives Tollens test but not a Fehling's test. So it is a wrong statement. Acetaldehyde gives a positive Fehling's test and a Tollens test, correct. Any aldehyde can give except benzaldehyde. Formic acid gives a positive Fehling's test and Tollens test. You see, formic acid, it is going to be like this. And this part of this structure looks like an aldehyde. It can give positive Fehling's test and a Tollens test, correct statement. And acetone gives a positive iodoform test and 2,4-DNP. Acetone is having CH3, C double bond O, CH3. So as this is having a methyl ketonic structure, it is going to give this test. So I want to say this statement correct, statement correct. Statement 1 is a wrong statement. It is incorrect. Option 1 is the correct answer for this question. Here it is a one question, which of the following will have the least hindered rotation about the carbon-carbon bond? Least hindered rotation means carbon to carbon bond can be easily rotated. That is possible in the case of ethane. In alkanes, carbon to carbon single bond can be easily rotated, while ethylene and acetylene dichloroethane also have the restricted rotation. And answer is going to be 1 for this question. Students, this question is based on valence bond theory postulates. Determine which of the following statement about VBT complex is incorrect. So there are 4 options. It does not explain the color of the coordination compounds. Correct. It explains only the magnetic behavior but not the color. It can distinguish between the strong and weak ligands. The entire valence bond theory is based on weak ligand and strong ligand, but VBT fails to explain which is a strong ligand and which is a weak ligand. And it does not explain the kinetic stability of the coordination compounds perfect, and it does not make the exact predictions regarding the tetrahedral and the square planar structures of coordination number 4 complexes. Most emphasis is given for the octahedral complexes only. And I would say structure 1, statement 1, 3, 4 are correct, and I feel statement 2 is incorrect. Option 2 is the correct answer for this question. Students, here is the route mapping conversions. So A is reactive with Grignard reagent and followed by hydrolysis. So you can take ethane nitrile CH3 C triple bond N. When it is treated with Grignard reagent CH3 MgBr followed by hydrolysis. So you are going to get CH3 C double bond O and CH3. This CH3 is given with the Grignard reagent. That is going to be, this is A and this is going to be R, and this R is treated with dilute NaOH. So when it is treated with dilute NaOH, you are going to get aldol condensation. This acetone, when it is treated with dilute NaOH and it is heated, it undergo aldol condensation. If you see aldol condensation reaction between these two, and it is going to be CH3 double bond and it is CH and C double bond O and CH3, and this is given as 4-methyl pent 3 into 1 is the correct structure. So that I can start with this ethane nitrile, and answer for this question is going to be option 3 is the correct answer for this question. Students, here it is a question, the incorrect statement among the following. So it is given 4 statements. Let us see. The first, ionization enthalpies of lanthanides are nearly 600 kilojoule per mole. So they have high ionization energies. If you see the NCERT table, the values are nearly close to the 600 kilojoules, which is a correct statement. And both neptunium and plutonium exhibit maximum oxidation state of the +7. In the NCERT textbook, graph is also given. They can show +7 oxidation state, which is correct. And the transition metal represents in Ziegler-Natta catalyst is titanium, titanium tetrachloride and triethyl aluminum is Ziegler-Natta catalyst. This is also correct. And europium has the smallest atomic radius. It is not having the smallest atomic radius, and which is actually a wrong statement. Among all, option 4, only D is the incorrect statement among these 4 statements. Students, here is a route mapping question from the organic chemistry. It is given as CH3, CH2, CH double bond CH2. The structure of A, I want to get hydroboration and oxidation. So anti-Markovnikov addition, alcohol is formed. So butane 2-ol is the answer for this structure A. And this is treated with alcoholic KOH. You are going to get again alcoholic KOH. You are going to get A again CH3, CH2, CH double bond CH2, alcoholic KOH. If it is treated with HBr, follow Markovnikov rule. When it follows Markovnikov rule, CH3, CH2 and CH and CH3 and Br is going to be formed, and this is going to be, this is going to be structure B. When it is treated with Br2 and CCl4, KOH, alcoholic KOH, when it is treated with alcoholic KOH, you are going to get elimination reaction CH3 CH double bond CH according to Zaitsev rule. And when it is treated with Br2CCl4, you will be getting CH3, CH and CH and CH3, and it is going to be Br and it is going to be Br. So Br2CCl4, and you use 2 moles of NaNH2. So double dehydrohallucination takes place. You are going to get CH3 C triple bond CCH3. I would say this is going to 2-butane alkyne is formed. Option 4 is the correct answer for this question. Students, this is asking about the complexes field strength. Which of the following complexes absorbs maximum wavelength in the visible region? So you have the maximum wavelength. So energy is inversely proportional to the wavelength. If the energy CFSE, crystal field stabilization energy, is more, and wavelength is going to be the minimum, and vice versa. If CFSE is less and wavelength is going to be the maximum, less CFSE means it happens with weak ligand. It happens with weak ligand. So among all, ammonia, water, cyanide and fluoride is the weakest ligand. So that I would say option 4 is the correct answer for this question. So students, here it is one more logical question. Benzene cannot react with. Benzene has 3 double bonds in it. Though it has 3 double bonds in it, it will not behave like an alkene, because delocalization of pi electron system above and below the ring makes benzene resistant to addition reaction. Therefore, Bayer's reagent, it cannot react. Br2CCl4 it cannot react, and HBr it cannot react. So I want to say it can react with Cl2Fe dark. Cl2Fe in the dark, you are going to get chlorobenzene. So I want to say option 2 A, B, C. Option 2 is the correct answer for this question. So here it is one question. The paper chromatography is a type of partition chromatography, and nitrogen present in the azobenzene can be detected by Lassine test but cannot be estimated by Zeldal method. Yes, it is correct. Whenever nitrogen present as a part of the ring and azo compounds, Zeldal method cannot be used. Therefore, both statements are correct. Paper chromatography is a type of partition chromatography. Based on that, in the paper GT paper 1 also one question has come, and more emphasis will be given on the practical approach of the chemistry for the NEET 2026. So both statement 1 and statement 2 are correct. Option 1 is the correct answer for this question. Student, here is the question based on the ether is treated with the ether, the major product B in the following reaction, hydrolysis of ether. When you hydrolyze this ether, you are going to get an alcohol and you are going to get an alkene. So what alkene you are going to get here? So when this is heated, SN2 reaction takes place. So it is going to be an alcohol and it is going to get an alkene. So carbocation is formed here, CH3 and CH2 and CH3, and it is going to be CH2 plus, and you will be having a carbocation rearrangement. So 1, 2 hydride shift. So as a result of this proton is going to move here, so that CH3, CH2, CH3 and CH3 plus, and this is deprotonated to gives rise CH3 and CH rule has to be followed, and CH3 and CH3. The alkene that is going to be formed, the most stable alkene, the answer for this question is going to be option 2. Students, this is general inorganic chemistry question. It is asking select the incorrect statements here: N2O5, CO2, B2O3, BeO, Li2O. It is a basic oxide, amphoteric oxide, weakly acidic oxide, and it is also weak acidic oxide. It is a very weak acidic oxide. It is an acidic oxide. So decreasing acidic strength, it is correct. Among Na plus and F minus, the smallest anion is going to be this much. Smallest ion is going to be. So as a negative charge cannot be isoelectronic negative charge. Isoelectronic negative charge should be bigger. So it is a wrong statement. Helium is used as a diluent for oxygen in modern diving apparatus because it has a very low solubility. It is said that very high solubility in the blood, which is not correct. And atomic size of yttrium is equal to lanthanum. Okay, this is not correct because of this lanthanide contraction. So B, C, D are the incorrect statements. I would say option 3 is the correct answer for this question. Identify the given reaction. There is a structure that is given reacted with SO2, SOCl2, thionyl chloride. Thionyl chloride is going to replace the hydroxy group with a chlorine atom, but not aromatic hydroxy groups. So this is aromatic phenolic. So this hydroxy group will not be reacted, only this and this will be reacted. So as a result of this, you will be having clear OH is intact and chlorine is replaced. For this question. It is option 4 is the correct answer for this question. Students, here it is a match the following question. List 1 and list 2 is given. Name of the reaction, Schwarz reaction, and you have to identify the product. In the Schwarz reaction, alkyl halide is converted into alkyl fluorides. So you can take ethyl fluoride, you can take an ethyl chloride. So A is going to be with 4. Sandmeyer reaction, benzene diazonium chloride, when it is treated with Cu2 Cl2, you are going to get a chlorobenzene, and Sandmeyer reaction can give you chlorobenzene. And Wurtz-Fittig reaction, in the Wurtz-Fittig reaction, alkyl halide and aryl halide react with sodium metal in the dry ether is going to give alkyl benzene, and you can say ethyl benzene, Wurtz-Fittig reaction. And Finkelstein reaction, alkyl chloride is going to react with sodium in dry ether, you are going to get alkyl iodide, you can take ethyl iodide. So these are the named reactions, these are the corresponding products. So answer option 3 is going to be the correct answer for this question. Students, here it is a one more assertion and reasoning type question. Assertion A, boiling point of alkyl chlorides, bromides and iodides are considerably higher than the corresponding hydrocarbon of comparable molecular mass. Of course, alkyl halides as the polar molecules as compared to hydrocarbons. It is a true statement. While intermolecular forces of attractions are stronger in halogen derivatives than hydrocarbons, that what I want to mention here. And in light of this statement, I would say both A and are true, and R is the correct explanation of A. Option 3, it is option 2 is going to be the option for this. Intermolecular forces, boiling point is directly proportional to intermolecular forces. Option 2 is the correct answer for this question. Students, here it is again assertion reasoning type question. Second ionization energy of iron is lower than that of third ionization energy of manganese. What is the meaning of third ionization energy of manganese? Manganese 2 plus become manganese 3 plus, for that require I3. Second ionization energy of iron, mean to say Fe2 plus become Fe3 plus, you require Ia2. Yes, IE2 of this is less than this because you have D5 configuration, you have D6 configuration. Assertion is correct. And Fe2 places D6 configuration and Mn2 places D5 configuration. Perfect. And reason is explaining the assertion. I just want to say both assertion and reason are correct, and reason is the correct explanation of assertion. Option 1 is the correct answer for this question. Again, we have both assertion and reasoning type question, bachcho. Phenol and benzoic acid can be distinguished by sodium hydroxide. You can't distinguish because phenol is acidic, it can react with sodium hydroxide, and benzoic acid is also acidic, it is reacted with sodium hydroxide. You can't distinguish, wrong. Benzoic acid is a stronger acid than phenol. Of course, carboxylic acids are stronger acid than phenolic compounds. So, assertion wrong and reason is correct. So, option 4 is the correct answer for this question. Statement based questions are more important in NEET examination. Stability of vinyl cation is more than allyl cation. I don't think so. Allyl carbocation, you see CH2 double bond CH and CH2 plus, it has a resonance. So, allyl carbocation is supposed to be more stable. And the IUPAC name of this 1, 2, 3, 4, 5, 6, 7, when double bond and triple bond is at equivalent positions, we have to check the next priority of this locant of the chlorine here. So, 3 chlorohept 6 ene and 1 ine is the correct answer. So, this is not correct and this is correct. My answer for this question is statement 1 is incorrect, but statement 2 is correct. Here it is a one more question. Which of the following vanadium dihalide is unstable? Vanadium is going to form dihalides with vanadium chloride, stable vanadium bromide, stable vanadium iodide, stable vanadium fluoride is the only unstable dihalide of vanadium. It is given in the NCERT. Option is going to be 4. Students, it is a question from practical organic chemistry, basic principles of organic techniques. So, 0.12 gram of organic compound gave 0.222 gram of magnesium pyrophosphate. By the usual analysis, the weight percentage of phosphorus, is going to be 62 by 222. Why? Because you are estimating in terms of magnesium pyrophosphate, its molecular mass is 62. In that magnesium pyrophosphate, 2 phosphorus are there, and weight of Mg2 P2O7 upon weight of organic compound into 100. So, 62 by 222. So, what is the weight of magnesium pyrophosphate? You got 0.222 upon 0.12 into 100. On doing so, you are going to get 51.6%. Option for 3 is the correct answer for this question. Students, this is a simple question. The element with atomic number 11, it is going to be Roentgenium, and in the periodic classification of elements, common names with atomic numbers are being given. You just pursue that, you are going to get, maybe you are going to get that kind of questions, very important. Students, here it is a question from amines. So, aniline is treated with CH3 acetic anhydride in the pyridine. You are going to get NH C double bond O CH3 acetyl N-acetylated aniline is formed. So, ring is now protected, group is protected, and it is going to react with Br2 and CH3COOH. Para-bromo product is the major product here, C double bond O CH3. Once it is hydrolyzed, you are going to get hydrolyzed. You are going to remove this to form NH2 and para-bromo product. When it is treated with amino group with NaNO2 HCl, diazotization takes place. NaNO2 and HCl, you are going to get N2Cl, and this is benzene diazonium chloride para-bromo. Now this treated with, this is D, and it is treated with copper bromide HBr, CuBr and HBr, and you are going to get Sandmeyer reaction, so Br, and it is going to be Br. So the answer for this question is para dibromobenzene, option 1 is the correct answer. So bachcho, overall the paper is going to be moderate and standard. In NEET examination, this is the type of questions you are going to expect. So we'll meet with more paper discussions in future. Thank you very much.
Hello everyone, good evening to all. So I'm here, Sumit. Solution for this grant test 2. Okay, so this is the instruction paper. You can read these instructions carefully. The instructions are same as for the final NEET exam. So I'm going to discuss the very first problem. You can see this, okay, from the physics part, okay. From the physics part, we will discuss the very first problem, okay. Let me go to that physics section. Yeah, and here we are. So the very first problem for question number 136, okay. It is a charged ball hangs from a silk thread S, which makes an angle theta with a large charged conducting sheet as shown in the figure. Okay, this is the ball here. Okay, B. Now this ball, on this ball, the forces acting are weight of it, which is mg. There is a tension in the string which is along the string. Okay, now this string is making an angle theta with the vertical. Okay, it is making an angle theta with the vertical. So you can take t cos theta to be mg. Okay, you can write t cos theta as mg. T cos theta is equal to mg. In the horizontal, there is another force which is the x component of the tension, and it is just t sine of theta. So you can write t sine of theta comes out to be, suppose charge on this ball is q, so q into e. Okay, so this is q into e. If you divide this equation by the second one, you are going to get tan theta is equal to qe divided by mg. Okay, now what that q is, okay, so q is the charge on this ball, e is the electric field. Now electric field is because of this conducting sheet P. Suppose it is having a charge density sigma, so you can write sigma by epsilon naught. So q sigma divided by epsilon naught divided by mg. Okay, that is equal to tan theta. Now if you see this expression carefully, this sigma, okay, q is a constant, epsilon naught is a constant, m is a constant, g is a constant, so sigma is proportional to tan theta. Okay, simple. That charge density is proportional to tan theta. Sigma is proportional to tan of theta. Okay, now the second one, 137 question. In an electric field region, the electric potential varies along the x-axis as shown in the graph. The x components of the electric field in the regions for the intervals pq and qr as marked in the graph, okay, as marked in the graph in Newton per coulomb are respectively. Okay, that is the question. This is second question, 137. So you can see in this region from origin to q, there is a line pq which is actually having a negative slope. Okay, how much slope that is? So if you see this angle, okay, if I calculate that slope, 1 second. Okay, let me write here. You can write, okay, actually I'm going to use this expression. E vector is equal to minus of del operator on V. This is for 3-dimensional, but because the potential given is 1-dimensional only, you can write E is equal to minus of dV by dx. What the dV by dx is representing? It is representing the slope. Okay, you can now see the slope is actually negative and slope is tan theta. Okay, tan theta. So this tan theta, if you see this diagram, comes out to be, okay, I'm calling this angle to be theta. Okay, so tan theta is equal to 5 divided by 1, 5 volt divided by 1 meter, so 5 volt per meter. Okay, that is actually electric field itself. With a negative sign we will put, okay, with a negative sign we will put. This is the value of tan theta. Theta is the angle shown here. Actually slope we take using this obtuse angle here. Okay, so you can just write the slope is minus of 10 theta, okay, that is minus of 5, okay, volt per meter. Now if you use this expression here, okay, this is actually the slope, this is actually the slope. You can write now this electric field is 5 volt per meter. Okay, 5 volt per meter, electric field is 5 volt per meter along the x direction. What about so this is 5.0 along positive x direction. Okay, so they are asking in the region PQ, in interval PQ and QR. Okay, QR is this. Here you can see now, let me again calculate the slope. Okay, so if I calculate the slope now, this is the line. Okay, we are talking about this line here. This is the line. Okay, now you can see I can write electric field as again that negative of dV by dx, or let me just write it as minus of delta V by delta x. Okay, if you see this region delta V, okay, final volt, okay, voltage at R it is minus 10, at Q it is 0. Okay, so minus 10 you can write with a negative sign, V2 minus V1 divided by x2 minus x1. This expression we can also use. So minus of this minus 10 here, minus 0, x2 minus x1. Okay, so this x2 is 1.5, x1 is 1, x1 is 1. So you can get 1.5 minus 1.0. Okay, now you are going to get minus minus. Okay, there will be a plus, so plus 10 divided by 0.5, and this is 20 volt per meter. Okay, again it is in the positive x direction. In both the cases it is positive x direction, 5.0 along positive x direction and 20.0 along positive x direction. So the right answer is the fourth one. Okay, fourth one. Also you can see actually electric field is from higher to lower potential. So this side higher potential, this side lower potential. So electric field is from higher to lower potential. Obviously it is along the x direction. Okay, calculation you can do just by calculating the slope. Now the next one, 138. Given below are two statements. One is labeled assertion A and the other is labeled as assertion R. Now assertion A is a point charge Q is placed at the center of a spherical cavity inside a spherical conductor as shown. Another point, okay, as shown. Another point charge capital Q is placed outside the conductor as shown. Now, as the point charge capital Q is pushed away from conductor, we are taking this capital Q charge away from the conductor. The potential difference VA minus VB between 2 points A and B within the cavity of a sphere remains constant. Yeah, that will remain constant. Cavity remains unaffected by the external effects, okay, by the external fields and the external charges. So this is a correct one. Okay, assertion is correct. Reason: the electric field due to charges on the outer surface of conductor and outside the conductor is 0 at all points inside the conductor. Okay, the electric field due to charges on the outer surface of conductor and outside the conductor is 0 at all points inside the conductor. Okay, this is also true. Okay, and it is the correct explanation. So both A and R are true and R is the correct explanation. First option is the correct one here. Now the next question, this is question number 139. Two heating coils, one of fine wire, okay, one of fine wire and the other of thick wire made of same material and of same length are first connected in series and then in parallel. Which of the following statement is correct? So you know resistance, if I write, resistance is resistivity into length divided by A. Fine wire, okay, one is fine, okay, so let me take resistivity is same for both of them, they are made up of same material. So rho L divided by A1, okay, cross-sectional area is less for the first one. Then R2 is equal to rho into L divided by A2. Now according to the question, this A1 is less than A2. Because A1 is less than A2, you can write R1 is greater than R2. Resistance of the thin one is greater than the resistance of the thicker one. Now, okay, they are first connected in series. So when you are connecting them in series, okay, they are going to have same current through them. They are going to have same current through them. Now because there is same current through both of them and resistance of the second, okay, resistance of the first one. Resistance of the first one is greater than the resistance of the second one. So So heat dissipation is more in the first one than in the second one if they are in series. Okay. So liberating more energy in the first one. If they are in parallel, in the case, okay, in parallel, okay, if they are in parallel with each other, here is R1, this is R2. So voltage difference is same. In this case, suppose H1 dash, this is the heat developed across the first one. H2 dash, this is the heat developed across the second one. So H1 dash is V square by R1, and this is V square divided by R2. R1 is greater than R2, so H1 dash is less than H2 dash. Okay, so in series, fine wire liberates more energy, while in parallel, thick wire liberates more energy. In series, fine wire liberates more energy, yes, correct. And in parallel, thick wire liberates more energy. So first option, the very first option is the correct one. Now this question, 5 equal resistances, each of resistance are connected as shown in figure. A battery of V volts is connected between A and B. The current following in AFCEB will be. Okay, so what I'm going to do, let me redraw this. Okay, let me redraw this. Okay, let me use, I'm redrawing it. So this is F point here. Okay, and okay, now between these 2 points, okay, here C, here it is D, okay, there is a resistor which is R, okay, there is a resistor which is R connected here, this is also R, this is also R, this is R, and this is R. Now it is a balanced Wheatstone bridge, okay, this is A, okay, and this is B here. You can call, okay, you can say this is F, you can call this is E, okay, and this is F, right? This is a balanced Wheatstone bridge, okay, balanced Wheatstone bridge. Now because it is a balanced one, you can remove this resistor between C and D, okay, you can remove this between C and D, this resistor. Okay, so if a battery of potential V is being applied between A and B, which path we are talking about? A, F, C, E and B. So current through this, okay, along this, here, and okay. So you can see in this path, okay, I hope this path is very clear to everyone, or let me redraw that here. Okay, so this kind of circuit we are getting actually. This is A, okay, then FCB, okay, A, F, this is C here, okay, and this is E, I think, okay, E here, and this is B here. So I'm talking about this current which is going through this, okay. So between F and E, there is a potential which is V, okay. So current is equal to, current I is equal to V divided by 2R. This is R here, this is R here, okay. So V divided by 2R, that will be the current. So V divided by 2R, okay, that must be the answer here. So the right answer is third option, okay, third option is the correct one. Now see the next question, 141. In the given circuit diagram, all the bulbs are identically rated, okay. Statement 1, if lamp B or C fuses, then light emitted by lamp A decreases. So if B fuses or C fuses, what will happen? Because they both are in parallel and they both are identical, okay. So if one fuses, resistance of the circuit will increase, you know. Initially there are two, okay, and they two are in parallel. So their equivalent resistance, if I write, that equivalent resistance is half of one of them, right? Now you are removing, so resistance will increase, it will just remain capital R if I only talk about, okay, this part in which B and C are connected. Okay, now because resistance is increasing, main current, okay, current through this circuit, it is going to decrease. Now because it is going to decrease, power consumed by the bulb, so the light emitting, it is going to decrease, as well as the potential through A is going to decrease. Okay, so statement 1, if lamp B or C fuses, then light emitted by lamp A decreases, correct. Statement 2, if lamp B or C fuses, then voltage on A decreases, this is also correct. Both the statements are correct. Okay, so the right answer is both statement 1 and statement 2 are correct. This is the right option. Now the next question, two identical wires A and B have the same length L and carry the same current. Wire A is bent into a circle of radius R. Okay, length L, radius R I want to find out. So obviously the circumference is equal to 2 pi R. Okay, so 2 pi R is equal to L. Question number 1 for 142 I am writing. This 2 pi R is equal to L. Radius is equal to L divided by 2 pi. Magnetic field at the center of it, it is mu naught I divided by R divided by 2R, or you can write B1 is equal to mu naught into I divided by 2 divided by this L by 2 pi. So you are going to get this B1 is actually equal to mu naught into pi, okay, into I divided by L. ये होगा magnet, this is the magnetic field. Now if B2, okay, so wire A is bent into a circle of radius capital R and wire B is bent into the form of a square of side A. Okay, so suppose you have a square of side A. Okay, current is the same from both of them. Okay, same current. So now the side of this square, if you write that side of the square, 4A, 4 times side of the square is equal to L, so one side is equal to L by 4. Okay, now because of one wire, okay, because of one wire, magnetic field I can find out. Okay, suppose this current I is flowing in this square. Okay, now these angles subtended are, okay, for this is 45 degree, this is also 45 degree, right? So you can write magnetic field due to 1 B. This is mu naught I divided by 4 pi D, okay, sine 45 degree plus sine 45 degree. Okay, so 2 by square root of 2, so B is equal to mu naught into I divided by this 4 pi. Now this D, actually this D of the center, it is half of the side. Okay, half of the side. Side is A, so this D is equal to L by 8. Now I can write mu naught by I 4 pi into L by 8. Here you can write 2 divided by square root of 2. Okay, now you can write this B is equal to, okay, this is 2 here. Then, okay, you will get mu naught, 2 mu naught into I. So you can get actually 4 mu naught into I. Okay, you can get 4 mu naught into I, 4 mu naught into I divided by square root of 2 pi L. 4 mu naught into I divided by square, oh, just 1 second, 4 mu naught into I divided by square root of 2 pi L. You can also write this total magnetic field is 4 times of it. If you make 4 times of it, 16 mu naught I divided by square root of 2 pi L. Now the ratio of them, ratio of B1 to B2. So if you divide B1 by B2, okay, let me do here itself. This B1 by B2, if you do, mu naught I, that will be cancelled out. Okay, L will be cancelled out. So you are left with pi divided by 16 divided by square root of 2, okay, divided by pi. So this comes out to pi square divided by 16 square root of 2. Now this 16 square root of 2, okay, so you multiply here square root of 2 divided by square root of 2. That operation I am doing only in the denominator. Okay, so 2 8 square root of 2. So pi square divided by 8 divided by square root of 2. This will be the answer. So the answer is second option. Okay, second option will be the correct 1. Now, if a charged particle of charge, if a charged particle of charge to mass ratio alpha enters in a magnetic field of strength b at speed v is equal to 2 alpha d into b. Okay, v is equal to 2 alpha d into b. What will happen if it is entering from this side? It will take a circular path. Okay, so it is going straight, right? This is this is the velocity, the index finger along the magnetic field. So thumb, that will give the direction of force. Okay, so v cross b. Okay, v cross b. So force is acting actually in the upward direction. So it will go like this. Okay, and come back. So semicircular path it will follow. Now this semicircular path will have a time period. We know that time period t is equal to 2 pi m divided by qb. Now according to the question, this m by q is alpha. So you can write 2 pi into alpha divided by b. For half of the time period it is inside. So that time period will be pi alpha by b. Okay. The ratio, charge to mass ratio, okay, not m by q, it is q by m. So alpha is here in the denominator. So time period is 2 pi divided by alpha b. For half of the time it will remain inside. So pi divided by alpha b, this is the time. Okay, pi divided by alpha b is the time. So angle subtended by the path of charged particle in magnetic field at the center of circular path is 2 pi. No, it is wrong. Okay, it is incorrect. That will be actually pi. Second, the charge will move on a semicircular path and then will come out from magnetic field at some distance from the point of insertion. So suppose this is here. This is the point of insertion here. From here it is entering and from here it is coming out. Okay. So the charge will move on a semicircular path and then will come out from a magnetic field at some distance from the point of insertion. This is correct. This is the correct option. Okay, this is incorrect because time is half of it. Angle subtended by the path of, okay, that angle is actually pi. Okay, semicircle is going to subtend pi angle. Now 144, the given figure represents a material which is, so lines are away, density at the place of material, lines are away, density is decreasing. So the material is actually, okay, that is actually repelling. So it is diamagnetic. Now what is the mutual inductance of a two-loop system as shown with center separation L? Okay, center is separated by L. So you know that magnetic field on the axis of a ring at a distance x from its center, if I write, okay, that magnetic field is mu naught into I divided by 2, okay, divided by r square plus x square raised to the power 3 by 2, okay, into r square. Okay, r is the radius actually. This is the formula I am writing. If you put x equal to 0 into it, okay, r cube you are left with, then mu naught I by 2r. Okay, in this way you can also check whether formula is correct or not. So magnetic field, now the radius is given as a, and I want to find out the magnetic field at the center of it. Okay, at the center of it, at the center of the second coil, because of the current in the first coil. So you can write this magnetic field comes out to be, okay, this is for question number 145 I'm doing, okay, this calculation, okay, for 145. So B is equal to mu naught into I into small a square divided by 2 divided by small a square. Separation is L, so in place of x I am writing L square raised to the power 3 by 2. Now this separation L is much greater than A, so you can neglect this A square. Now you are going to get mu naught into I into A square divided by 2 divided by A cube. So mu naught I, okay, no, you are going to neglect, because L is much greater than A, so you will neglect A square, not L square. You can now write 2, and if you neglect it, you are left with L square, cube root, okay, square root of L square is L, so L cube. Mu naught I A square divided by 2L cube. This is the magnetic field. Now this magnetic field, because it is at a very large distance, so I can assume that along the okay, along the surface of the second ring, this magnetic field is same, okay, and normal, okay. Here at the center, it is normal to the plane, okay. Because it is much far away, I can assume that magnetic field in the plane of the second coil, okay, for all the points in the second coil remains same and is normal. Now you can write the flux through it. If you write the flux phi, that flux phi is equal to this magnetic field into area of it. What that area is pi a square. So you can multiply by pi a square. So multiply it with pi a square. This comes out to be mu naught into I into pi into a raised to the power 4 divided by 2 divided by L cube. If you want the mutual inductance, you just divide this phi by current. So mutual inductance M is phi divided by the current. If you divide phi by the current, you're going to get mu naught pi a raised to the power 4 divided by L cube. Mu naught pi a raised to the power 4 divided by L cube. Mu naught pi a raised to the power 4 divided by 2L cube. Fourth option is the correct one. Now an 8 microfarad capacitor is connected across 220 volt 50 hertz line. What is the peak value of charge through capacitor? So the peak value of the charge that is just equal to capacitance into maximum value of voltage. Okay, so you can write, just let me go back, do the calculation here. So you can write Q, this charge Q, maximum charge Q is equal to C into maximum voltage. If you see the C, okay, where is that C? Okay, capacitance, it is 8 microfarad, 220 into square root of 2. Okay, so 8 into 8 microfarad, 220 into square root of 2. Calculate it. Okay, so if you calculate, this is 1.4 into 0, that is 28. So 28 into 22 into 8. Okay, so this comes out to be nearly, okay, they are asking in, fine. If you write for microfarad, that is 10 raised to the power minus 6. Multiply them. 22 into 8, that is, okay, so 17, okay, 44 into plus 17, that is 61 into 8. Okay, this will be the, okay, so an 8 microfarad capacitor is connected across 220 volt. Okay, let me further check. 8 into 220 into square root of 2. Okay, so 20, fine. 8, this is the square root of 2, that is 1.4 into 0, this is 14. 14, okay, 14, okay, this is 14. Okay, this will give me only 14, not 28. So I'm getting double the answer actually. 14, you can now write this as 14 into 8 into 22 into microfarad, that is 10 raised to the power minus 6. Okay, if you multiply now, (2464) into 10 raised to the power minus 6. So 2.464 into 10 raised to the power minus 3 coulomb. This is the answer. 2.464 into 10 raised to the power minus 3 coulomb. So the right answer is here. Okay, this is the most appropriate answer. 2.5 into 10 raised to the power minus 3. Now the next question is, an AC source of angular frequency omega is fed across resistor R and capacitor C in series. The current registered is I. If now the frequency of source is changed to omega by 3, the current in the circuit is found to be halved. calculate the ratio of the reactance to resistance at the original frequency. How we can do that? So an AC source of angular frequency omega is fed across resistor R and capacitor C in series. So first we have this there is a resistor, there is a capacitor, both are in series, resistor and capacitor. Connected with the source of angular frequency omega. Now you can write this Z is equal to square root of R square, okay, R square. Now XC, this is equal to 1 divided by omega C, okay. So 1 divided by omega square C square, okay, that is Z. This is Z. Now if you write the current, that current I, okay, is equal to, suppose this is the EMF E applied, so E divided by this Z. Now according to the question, current becomes half. When the current becomes half, current becomes 1/3 if the frequency is changed to, current becomes half. The current in the circuit is found to be halved if frequency is being changed to omega by 3. So if you change the frequency to omega by 3, that current becomes I by 2. So EMF remains same. Here you can write impedance, that is R square plus 1 divided by, okay, in place of omega, if you write omega by 3, you will get omega square by 9 actually. So I'm directly writing here 9 divided by omega square C square. That you are going to get. If you put both of them equal, okay, if you put both of them equal, you are going to get this E divided by square root of R square plus 1 divided by omega square C square. That is equal to E divided by square root of R square plus 9 divided by omega square C square. Okay. If you make square of both sides, you are going to get R square plus 1 divided by omega square C square. Okay. Also 1 by 2 is here. So they both are not equal actually. Okay. This expression, this, where is this one? Okay, E by Z. So here I am writing E by Z. Okay, this is I. Okay, this is I. And here, from here, if I write I, 2 I have to multiply with. Okay. Now if we make a square of both sides, so E and E cancelled out and you are left with R square plus 1 by omega square into 4, that is equal to R square plus 9 divided by omega square C square. Now 4R square minus R square, this is 3R square you can get, and 9 by omega square C square, okay, you can get 5 divided by omega square C square. Okay. 5 divided by omega square C square. Right? Now what the question is? The question is calculate the ratio of reactance to resistance. So reactance is 1 by omega C. Okay? 1 by omega C. So 1 by omega C we are getting XC. Okay, so this is the reactance at the original frequency. So 3R square is equal to 5 into XC square. So R square divided by XC square is equal to 5 by 3. That is R by XC is equal to square root of 5 by 3. Okay, this is square root of 5 by 3. Square root of 5 by 3 is the right answer. Square root of 5 by 3. Okay, so they have given reactance to resistance. Okay, so take the reciprocal of it. Reactance to resistance. Square root of 3 by 5. Okay, square root of 3 by 5, that is the second option and it is the correct one. 147. Now, the magnetic field in the plane electromagnetic wave is given by this expression. Okay, there is an expression written here, Bz is equal to 2 into tan raised to the power minus 7. Sine of 0.5 into tan raised to the power 3x plus 1.5 into tan raised to the power 11t. And we are writing obviously this in Tesla. Now the expression for electric field is, so you can see maximum value of electric field is given. This is 2 into tan raised to the power minus 7. What is, okay, what is now the electric field? Okay, electric field is just equal to Bj. Okay, electric field is just C times of B. Maximum value I am just talking about. Okay, so this maximum value is 2 into 10 raised to the power minus 7. So you can write 3 into 10 raised to the power 8. This is the speed of light. B 2 into 10 raised to the power minus 7. So this comes out to be 60 into 10, which is 60 volt per meter. If you write the equation, okay, so you can use, okay, actually you can write the relation in vector form. Okay, if you write in the vector form, this is E vector is equal to C times, okay, C times B cross K. Okay, what is K? K is the propagation direction of the wave, okay, of this electromagnetic wave. Now you can see it is K. Okay, if I make a comparison with Kx omega t, okay, argument, if you write the magnetic field B as B dot sine of Kx plus omega t, it is actually moving in the minus x direction. Okay, minus x direction. So minus I cap cross, okay, minus I cap, that is the direction of wave propagation. This is the direction of this K vector. Magnetic field is in the z direction. Okay, so minus of z cap cross I cap. Z cap, okay, K cap cross I cap, you can say. So K cap cross I cap, that is minus y cap. Okay, K cap cross I cap, K cross I cap, that is actually, okay. The expression for electric field they are asking, just 1 second. Magnetic field B is equal to 1 by omega into K cross E. If I take a vector product with K cross B, okay, so K dot E into K dot E into K minus K dot K into E. So electric field, obviously that is in the opposite direction of K cross B, that is along, okay, it is parallel to B cross K. B cross K. K is the direction, K vector is along the direction of wave propagation. So from this we can say it is along the negative x. Okay, negative x, not x. So minus I should take. Minus I cap. Okay, 1 second. I want to find out the direction of, so B is along Z direction, so K cap. This K vector, okay, which is along the minus X direction, so K cross I cap minus with a minus sign. Now K cross I cross J is K, okay, J cross K, that is I cap, K cross I, it is minus J cap, so along the positive, okay, sorry, K cross I cross J is K, J cross J cross K, it is I cap, and K cross I, this is just J cap. Okay, so negative J. So EY, expression of EY, that should be 60 sine of 0.5 into 10 raised to the power 3x plus 1.5 into 10 raised to the power 11t. So I can find actually two options are same here. Okay, the expression of electric field. So two expressions I'm finding same, two answers. Second one and the fourth one. Okay, so two options are same actually. Now, a convex lens is dipped in a liquid whose refractive index is equal to the refractive index of the lens, then its focal length will be. So focal length, for the focal length you can write the formula that focal length 1 by FL is equal to refractive index of the glass divided by refractive index of the liquid minus 1 into 1 by R1 minus 1 by R2. Now okay, because refractive index of the glass is equal to refractive index of the liquid. So mu G by mu L is 1. Okay, so 1 by FL comes out to be 0. Because it is 0, you can write FL is equal to 1 by 0, which is actually infinite. Okay, this is infinite. So infinite, it becomes infinite. Next question. Next question is 150. A small coin is resting on the bottom of a beaker filled with a liquid. A ray of light from the coin travels up to the surface of the liquid and moves along its surface. Because it is moving along the surface, that means it is falling at an angle which is equal to critical angle. It is falling at an angle which is equal to critical angle. This is the critical angle theta c. Okay. Now because this is theta c, this is also theta c. Okay. Now you can write this. What you can write? Sine theta c, from this diagram, you can write sine theta c. It is perpendicular divided by the hypotenuse. Hypotenuse will be, okay, if you see this is A here. Suppose this is A here, this is B here, and this is C here. Okay, so sine theta c is equal to perpendicular divided by hypotenuse. Hypotenuse is AC and it is equal to 5. So sine theta c is equal to 3 by 5. Sine theta c is equal to 3 by 5. Now you can write, refractive index. Okay, sine theta c is also equal to 1 divided by refractive index of the denser medium, okay, with respect to refractive index of the rarer medium. Okay, now sine theta c we have calculated, this is 3 by 5. Okay, this is 3 by 5. Just one second. I will write mu 2 sine theta c is equal to, mu 2 sine theta c is equal to 1. Okay, so that is just 1 by mu 2. Let me write just 1 by mu 2 here. It is air outside. Okay, I am assuming it is air. Okay, so sine theta c is 3 by 5. Now this is 1 by mu 2. Mu 2 comes out to be 5 by 3. 5 by 3 is the refractive index of that liquid. So the speed of light is just 3 into 10 raised to the power 8 divided by 5 by 3. So speed of light is 3 into 10 raised to the power 8 divided by this 5 by 3. It comes out to be 9 by 5. 9 by 5 into 10 raised to the power 8, which is 1.8 into 10 raised to the power 8 meter per second. So 1.8 into 10 raised to the power 8 meter per second. This is the answer. Now see the next question. In a YDAC light of wavelength lambda equal to 5000 angstrom is used, which emerges in phase from two slits a distance d apart. A transparent sheet of thickness t of refractive index mu is placed over one of the slits. Where does the center maxima of the interference now appear? So if we see this diagram here, theta, okay, this theta angle is given. Okay, they have given this. Okay, you can see in this diagram, this is the theta angle. Theta, if we assume that the separation between the plane of the slits and this screen, it is sufficiently large, theta is very small. So you can take, when theta is small, sine theta is approximately equal to 10 theta. So that path difference between the two rays emerging, S2P minus S1P, if I write S2P minus S1P, it comes out to be equal to d sine of theta. Okay, this is d sine theta. S2P minus S1P, that comes out to be d sine theta. Now sine theta can be taken as, suppose this is y here, okay, and this is capital D, so this is y divided by capital D. Now you can write this is d into y divided by capital D, small d into y divided by capital D. This is the phase difference between the, this is the path difference between the two waves, one emerging from S1, the second one from S2. Now if I, okay, suppose we introduce a slab, okay, where, suppose we introduce a slab here, opposite to S1, right? So now the effective path, okay, effective path for the ray which is emerging, for the wave which is emerging from S1, that will be equal to, you can write, it will be equal to S1P minus of the thickness of the slab plus the optical path, okay, which is mu t. Now you can write S1P, you take t common, into mu minus 1, okay, S1P plus t into mu minus 1, okay. Now if you take, where is that, S2P minus S1P. So effective path difference, okay, effective path difference now will be less than by, okay, so effective path difference, let me write delta x, it is equal to now d sine theta minus of t into mu minus 1, okay. So d sine theta minus t into mu minus 1, it should be equal to n lambda. It should be equal to n lambda. What this d sine theta is, this is d small d into y divided by capital D minus t into mu minus 1, and this is equal to n lambda. Okay, now you can write this dy divided by capital D is equal to t into mu minus 1 plus n lambda. Okay, now you can get the position of the central fringe by keeping n equal to 0. So if you keep n equal to 0, so small d into y divided by capital D, this comes out to be t into mu minus 1, and you can get the position y is equal to t into mu minus 1 into capital D divided by small d. Okay, t into mu minus 1 into capital D, t into mu minus 1, t into mu minus 1 into capital D divided by small d. That is fourth option is the correct one. Next, the maximum intensity in Young's double slit experiment is I naught. Distance between the slits is d equal to 5 lambda, where lambda is the wavelength of monochromatic light used in the experiment. What will be the intensity of light in front of one of the slits on a screen at a distance capital D equal to 10 into small d? Okay, so what will be the intensity of light? So distance between the slits is 5 lambda. Now what will be the intensity of light in front of the slits on a screen at a distance capital D equal to 10 into small d? So before this, let me draw a diagram. Okay, let's just make a diagram. These are the two slits. Okay, here is the screen. Now just before one of the slit, I want to find out, okay, at this point the intensity. This is question number 151. Okay, so this distance is actually small d by 2, right? So the question is, what will be the intensity of light in front of one of the slits on a screen at a distance capital D equal to 10 into small d? Capital D is equal to 10 into small d. Small d according to the question is equal to 5 lambda. I can find out the path. Okay, at this point there are two waves reaching, one from this slit, it's okay, and one from this. What will be that path difference? Okay, so that path difference, always remember, you can remember that path difference, or you just write d sine theta. Always remember that if I join this point with the center of the slits, then that angle is theta. Okay, and you can easily estimate, okay, this angle is actually theta. So path difference is just small d into y divided by capital D. This is the path difference. Y is equal to, okay, that position is d by 2. So y is equal to d by 2. So d into small d by 2, capital D, that this is 10 into small d, and small d cancelled out. Okay, now you are going to get d small d divided by 10. This is small d divided by 10. Small d is equal to 5 lambda. So you can write 5 lambda divided by 10. This comes out to be lambda by 2. Okay, lambda by 2. If you write from this the phase difference, delta phi, this is equal to 2 pi divided by lambda into lambda by 2. Lambda and lambda cancelled out. This is pi. Okay, you're going to get pi only. Okay, let me once check again. So d into capital Y divided by d small d into y divided by capital D, small d, okay, y, okay, this is small d by 2. Okay, d is itself equal to 5 lambda. Okay, so d and d cancelled out. There will be a 20, not 10. There will be a 20 here. Okay, so lambda by 4 I am going to get. Okay, lambda by 4. Lambda by 4, this is the path difference. So phase difference, this comes out to be now pi by 2. Pi by 2. Okay. So the maximum intensity, if it is I naught, when there will be a maximum intensity. The maximum intensity, okay, maximum intensity. If this source is producing an intensity I, this is also I. So I max is I plus 2 into square root of I into I. This comes out to be 2I plus twice of I, that is 4I, which is I naught according to the question. Okay. Now if this is pi by 2 is the phase difference, okay, so in that case the intensity at point P is equal to I plus twice of square root of I into I cos pi by 2. Cos pi by 2 goes to 0, so you get 2I. Okay, 4I is equal to I naught, 2I is equal to I naught divided by 2. Okay, so I naught divided by 2 is the answer. First option is the correct one. Now in a photocell, bichromatic light of wavelength 2475 angstrom and 6000 angstrom are incident on cathode whose work function is 4.8 electron volt. Now if a uniform magnetic field of 3.3 into 10 raised to the power minus 5 Tesla axis parallel to the plane of plate, the maximum radius of the path described by the photoelectron will be. How we can find out that? First we need to find out what is the maximum velocity possible. Okay, so these are the two wavelengths given, 2475 and 6000 angstrom. Okay, 2475 and 6000. Let me write here, lambda 1 is equal to 2475 angstrom. Lambda 2, it is equal to 6000 angstrom. Okay, now you can write this maximum kinetic energy, suppose it is K1, is equal to hc by lambda 1 minus the work function. Okay, hc by lambda 1. Okay, so you can write it as 1242 electron volt nanometer. Okay, 2475 angstrom is there. Okay, so how many number of, okay, 247 angstrom, so 1242 electron volt nanometer, right? I am writing, so if I convert it into nanometer, that will be 247.5. This is hc by lambda 1. What is the work function given? That work function is given as 4.8 electron volt. Okay, so if I divide it, okay, okay, 247.45. So this is almost, if I multiply it by 5, this quantity, okay, so it comes out to be nearly 5. Okay, nearly 5. If I say for 6000, if you put 6000 here, work function now you can see is equal to 4.8 electron volt. So this 6000, if you convert, if you see the corresponding energy of 6000, so 1242 divided by 600, that is nearly 2. So second radiation is unable to emit the electron. Okay, so we can leave that. Okay, now come to this only 2475. Okay, so 24.75, this is so if I calculate it, okay, so 1242 0 divided by 2475. So if I do this calculation exactly, a 5, okay, Okay, then 0 here. Okay, so 5.02 electron volt. Okay, this is the energy of the radiation falling. Now you can write kinetic energy possible, maximum kinetic energy possible. This comes out to be hc by lambda minus the work function. Hc by lambda is now 5.02. Work function is 4.80 electron volt. So 2 here, okay. Then 0.22 electron volt. This is the energy of the this is the maximum energy of the particle. Now, maximum radius. So radius is equal to mv by qb. mv by qb. Okay, mv is just the momentum. In place of it, I can use the energy expression, square root of 2 mk. 2 mk divided by qb. Now, if I put all the values, square root of 2 into 9 into 10 raised to the power minus 31. In place of k, you have 0.22. 0.22 into 1.6 into 10 raised to the power minus 19. Okay, then you have here q, which is also 1.6 into 10 raised to the power minus 19. There is magnetic field, and that magnetic field is 3 into 10 raised to the power minus 5 Tesla. So 3 into 10 raised to the power minus 5. Okay, so okay, this will also give me a square root 10 raised to the power minus, okay, you can get a square root of 2 into 9 into 10 raised to the power minus 31 into 0.22 divided by this square root of 1.6 into 10 raised to the power minus 19. Okay, divided by 1 by 3 divided by 10 raised to the power minus 5. So what we are going to get? Square root of 2 into 9 divided by 1.6, okay, 10 raised to the power minus 19, this will give you 10 raised to the power minus 12, okay, into 0.22, okay, divided by 3 divided by 10 raised to the power minus 5, okay. So 0.22, 1.6, 10 raised to the power minus 6, what we are going to get from this? 0.22, okay, that will give me nearly 8. Then 4, 9 by 4, this will give me 3 by 2. So 3 by 2 into 10 raised to the power minus 6 divided by 3 divided by 10 raised to the power minus 5. 3 and 3 cancelled out, 1 by 2 into 10 raised to the power minus 1 meter. Okay, so this is nearly 1 by 2 into 10 raised to the power minus 1 meter. You multiply it by 100. Okay, then 10 by 2 centimeter. So 5 centimeter nearly. That will be the answer. Okay, second option will be the correct one. Now the next question. The figure shows a plot of photocurrent versus anode potential for a photosensitive surface for 3 different radiations. Which one of the following is a correct statement? So the very first statement for this, okay, this is the graph given here. Curves A and B represent incident radiation of different frequencies and different intensities. Now you can see, okay, retarding potential is same for A and for B also. Okay, so you can write EVA. This is equal to HC divided by lambda A minus work function. Then EVB, that is equal to HC by lambda B minus the work function. Okay, EVA, this is HC by lambda A minus work function. EVB, this is HC by lambda B minus work function. Now you can see potential VA and VB, they both are same. Okay, so I can say lambda A and lambda B are same. That means frequencies are same. But the magnitude of current, you can see, as we are increasing the potential, okay, as we are increasing the anode potential, retarding potential is that potential which is stopping, okay, at that potential on the anode, no electron is reaching the anode. So if this current you increase, okay, if this retarding potential you increase, anode potential you increase, current is increasing, that means intensity of B is more than the intensity of A. So right option is, let me check one by one. The very first option they have given, curves A and B represent incident radiation of different frequencies and different intensities. Curves A and B represent incident radiation of different frequencies and different intensities. It is incorrect. Curves A and B represent incident radiation of the same frequency but of different intensities. This is the right option. Okay. Now, an orbital electron in the ground state of hydrogen has an angular momentum L1, and an orbital electron in the first orbit of the ground state of lithium has angular momentum L2. We know from Bohr's law that in the nth orbital, okay, angular momentum is just given by NH cross. Okay, it is same for all the atoms. So L1 is equal to L2. That is the right answer. Next question, a radioactive nucleus undergoes alpha emission to form a stable element. What will be the recoil velocity of the daughter nucleus if V is the velocity of alpha particle and alpha emission, and A is the atomic mass of radioactive nucleus? Okay, so the mass of the alpha particle, you can write mass of the alpha particle, what you are considering. Mass of the alpha particle. Okay, let us take just 4 times of mass of proton. Mass of the nucleus remaining. Okay, they are asking what is the recoil velocity, what will be the recoil velocity of the daughter nucleus. So the mass of the daughter nucleus, if you write after emission, okay, let me write it as md dash, it is equal to A minus 4 times of mass of proton. Mass of proton and mass of neutron, they are almost similar. So if you write the initial momentum, that initial momentum is 0. Now you can write m alpha into V alpha plus md dash into Vd, it comes out to be 0. Now you can write this recoil velocity Vd is equal to minus of m alpha divided by md dash into V alpha. What is this m alpha? That is 4 times of mass of proton. For daughter nuclei, it is A minus 4 times of mp. So you are going to get mp. Okay, you are going to get this 4 divided by A minus 4. You are going to get 4 divided by A minus 4 into velocity of alpha, that is V. Okay, you can put negative sign, but we want only the magnitude of it. So 4b divided by A minus 4. Now in the circuit shown in the figure, the maximum output voltage V0 is, okay, so maximum output voltage we are taking across this resistor connected, okay, across this resistor connected here we are taking. Now when the positive half cycle is there, okay, when it is maximum, when obviously when the maximum positive voltage is there, will be there, right? So it can happen, okay, what is the maximum output voltage V0 they are asking? Okay. So suppose during the positive half cycle, D2 remains forward biased while D1 remains reverse biased. Okay. During the negative half cycle, D1 will conduct, D2 will not conduct. But the potential drop, okay, be it in the positive cycle, be in the negative cycle, that maximum positive voltage will be same. The polarity may differ. Okay. So I am just assuming for the first positive half cycle. Okay. In that case, the equivalent diagram will look like, okay, as I am drawing here. Okay, then this is the current, and this is the current here. Okay, right, here you are applying the voltage. This is the 10 volt. So you can consider actually there will be a 10 volt. Okay, there will be 10 volt of potential here. Let me name them. Okay, suppose this is A here, this is B here, right? So actually I want to find out potential drop here, okay, across these two. Okay, suppose this is A here and this is B here, right? This is A, this is B here. Okay, now if you see, I can say that there is a potential difference of 10. Okay, there is a potential difference of 10, across this 2 kilo ohm and this 2 kilo ohm. Okay, let us take this point as C. So between B and C there are two resistors of values 2 kilo ohm and 2 kilo ohm connected. Okay, so I can write the current from B to A to C. Okay, the current from B to A to C I is equal to, okay, that is equal to 10 divided by 4 kilo ohm. That is equal to 10 divided by 4 kilo ohm, or it is equal to 2.5 milliampere. 2.5 milliampere. Now the output is being taken for the resistor connected between A and B. You multiply it by 2 kilo ohm. So that voltage comes out to be 5.0 volt. This is the maximum output voltage. 5.0 volt. 5 volt. Okay. Now the next question 158. In figure, the current supplied by the batteries, diodes are ideal. So if you see the polarity of the battery as shown in this diagram, the upper diode, it is forward biased, lower one is the backward, okay, it is the reverse biased. So current will take this path, okay, go like this, okay, then come back. So 20 ohm is the resistance here, 2 volt is the voltage. Now you can write current is actually equal to 2 volt divided by 20 ohm, and this is 0.1 ampere. 0.1 ampere will be the right answer. That is option 1. Choose a false statement from the following. Substances with energy gap of the order of 10 electron volt are insulators. Okay. So yeah, that is correct. Those are insulators. The conductivity of a semiconductor increases with increase in temperature. This is also correct. In conductors, the valence and conduction band may overlap. Okay. And the fourth one is the resistivity of a semiconductor increases with the increase in temperature. So the false one is fourth one. Okay. Fourth one is the correct answer here. Now the next one, if C, V, L and I are capacitance, voltage, inductance and electric current respectively, then the dimensions of CV square by Li square is. We know that, okay, CV square, that is 1 by 2 CV square is the potential energy stored in a capacitor if voltage V is across it. 1 by 2 Li square is the potential energy, is the energy stored in the inductor. So both have the same dimensions, ratio is dimensionless, M0, L0, T0. This is the answer. Okay, 160. Okay, for 160th question, second option is the correct one. Now, a student measured the diameter of a small steel ball using a screw gauge of least count 0.001 centimeter. The main scale reading is 5 millimeter and 0 of circular scale division coincides with the 25 divisions above the reference level. Then you can write total reading. Okay, total reading is equal to main scale reading, which is 5 millimeters, so 0.5 centimeter. Okay, plus number of divisions into least count. Number of divisions, that is 25 into 0.001. Okay, so this comes out to be 0.5 plus 0.025. Okay, and it comes out to be 0.525 centimeter. Now, because there is a negative zero error, that means the measured value from this screw gauge is actually less than the actual result. You must add into it the magnitude of it, which is 0.004. So the total reading, to get the total reading, I will also add this zero error. Okay, so you are going to get your answer as 0.529 centimeter. Now the next question, a particle starts from rest with uniform acceleration and its velocity after n seconds is v. The displacement of the body in last 2 seconds is. Okay, so a particle starts from rest. Suppose that acceleration is a. Now you can write velocity after n second v is equal to an. Now the displacement of the body in the last 2 seconds. So suppose the displacement in n minus 2 seconds I write. Because it is starting from rest, that is 1 by 2 into a into n minus 2 whole square. If you write in n second, that is 1 by 2 into a into n square. If you take a difference, okay, that will be the distance traveled in the last 2 seconds. So 1 by 2 into a, okay, so Sn minus 2, so 1 by 2 into a, that is n square minus n minus 2 whole square. Okay, So if you solve it, you are going to get 1 by 2 into a, 1 by 2 into a n square minus n square minus 4, okay, and plus 2n. Let me write here plus 2n. This is the difference, and it comes out to be now 1 by 2 into a, 2n minus 4, 2 you can take common, so a into n minus 2, okay, a into n minus 2. In place of a n minus 2, in place of a, you can write v divided by n, okay, you can write v divided by n. So you are going to get v into n minus 2 divided by n, okay. Let us see now which option is the correct one. Okay, no one. Just 1 second, let me check it again. 1 by 2 into a into n minus 2 whole square, 1 by 2 into a into n square. If I take a difference, that is 1 by 2 into a n square minus n minus 2 whole square. 1 by 2 into a n square minus n square, okay, minus 2 square, that is 4. So minus 4, okay, then minus 4n. Minus minus, that will be a plus, so plus 4n you are going to have. Plus 4n you should write. Okay, so you can now write plus 4n. Now it is 2 into a into n minus 1 here, okay, so 2 into v into n minus 1 divided by n. That will be the answer. 2v into n minus 1, 2v into n minus 1 divided by n. That is the answer. Now the next question. Next question is, a wedge is placed on a smooth horizontal plane and a rat runs on its sloping side. The velocity of wedge is v equal to 4 meter per second towards right. What should be the velocity of rat with respect to the wedge u, so that the rat appears to move in vertical direction to an observer standing on the ground? Okay, standing on the ground. So you can write the velocity of rat, okay, velocity of rat with respect to this plank, okay, velocity of rat with respect to the plank URP, it is equal to u cos 60 degree with an I cap, okay, and negative sign, then u sin 60 degree j, u sin 60 degree j cap. Okay, now velocity of this plank, it is towards right, okay, so you can write velocity of this rat with respect to plank is equal to velocity of rat minus velocity of plank, okay. This velocity of the plank is actually vi cap, okay, so you can write urp is equal to minus u cos of 60 degree I cap plus u sin 60 degree of j cap, okay, and this is velocity of rat minus velocity of, okay, this plank, which is vi cap. Okay, now you can take up this minus vi cap towards right. Okay, so you can write velocity of the rat, velocity of rat is equal to minus u cos of 60 degree I cap plus vi cap plus u sin 60 degree j cap. Okay, now it's because that final one is in the vertical direction, x component must be 0. If x component is 0, minus u cos 60 degree plus v is equal to 0. V is equal to u cos of 60 degree. Okay, v is equal to u cos of 60 degree. V is given as 4. So 4 is equal to u by 2. This u comes out to be 8 meter per second. This is 8 meter per second. 8 meter per second. Now the next question is, when a particle moves in a circle with a uniform speed, its velocity and acceleration are both constant. No, it is not true. Okay, its velocity is constant, but the acceleration changes. This is also not true. its acceleration is constant. This is also not true, because acceleration, the direction of acceleration will also keep on changing. Okay, if it is moving with uniform speed, acceleration is always towards the center, and that radial direction is continuously changing. Okay, so fourth option, its velocity and acceleration both changes, is the correct one. Now, in the figure, if block A and wedge B will move with the same acceleration, then the magnitude of normal reaction between the block and the wedge will be. There is no friction between block and the wedge moves on horizontal surface. Okay, so suppose there is a normal reaction acting here. Let me just draw a free body diagram for A. So there is a normal reaction acting. There is its weight acting, okay, which is 2 mg. Okay, now what? Okay, this is the angle it is making with the vertical. Okay, and okay, what they are saying, that they both are moving, then the magnitude of the normal reaction will be when block and the wedge will be, okay, in the figure, if block A and wedge will move with same acceleration, A and B moves with same acceleration. Okay, suppose towards left they are moving. Okay, so this is 2 m mass here, this is 2 ma. Okay, so this 2 ma, okay, that is the 2 m is the force actually acting. Okay, acceleration is towards left, right? Acceleration is towards left. Fine. So if you see this, n cos theta must be equal to 2 mg and n sin theta must be equal to 2 ma. Okay, so you can write easily n cos of theta is equal to 2 mg. N cos theta is equal to 2 mg. Because they both are moving with the same acceleration, I assume, okay, there is no relative motion happening between them. Okay, so n is equal to 2 mg divided by cos of theta is the right answer. 2 mg divided by cos theta is the right answer. Now the next question, if W1, W2 and W3 represent the work done in moving a particle from A to B along 3 different paths 1, 2 and 3 respectively in the gravitational field of a point of mass m, find the correct relation between W1, W2 and W3. Because the motion is happening under gravitational field, which is a conservative field, so the work done along all the paths work done are equal. So second option is the correct one. Now the potential energy of a system is represented in the first figure. The force acting on the system will be represented by, okay. So you can see that potential energy is increasing with x and having a positive potential. Now force can be written as minus of du by dx. This straight line is going to have a positive potential and a constant one, so force is negative up to what point? Up to A. Force is negative. After this, because potential energy becomes a constant, force becomes a 0. Okay, force becomes 0. So third one, that will be the right option. Third option is the correct one. Now the next question, two uniform non-conducting balls A and B have two identical size having radius r but made of different density material. The ball A is positively charged and ball B is negatively charged. The balls are released on the horizontal smooth surface at the separation 10r as shown in figure. Because of the mutual attraction, the balls start moving towards each other. They will collide at a point. So where they are going to collide actually. Now let me write actually during the entire motion, center of mass will remain at its own position because all the forces are internal forces. So you can write center of mass position as m1 x1 plus m2 x2 divided by m1 plus m2. Okay, now if you see this m1, okay, radius is r for this A sphere, okay, radius is r for this B sphere. So two uniform non-conducting balls A and B have identical size having radius capital R but made of different density material. Density of A is equal to twice the density of B. Okay, so the ball A is positively charged and ball B is negatively charged. The balls are released on the horizontal smooth surface at the separation 10r as shown in this diagram. Okay, because of mutual attraction, the balls starts moving towards each other. They will collide at a point. Okay, so initially suppose, okay, this is the center of mass of the first one, okay, for A ball, and this is the center of mass of the second ball. Okay, so coordinate initially x1 is equal to 0, mass m1, that is equal to density into volume. So volume is 4 pi by 3. 4 pi by 3 r cube into rho. M2, this is equal to 4 pi by 3, okay, identical size 4 pi by 3 into r cube into 2 rho. Okay, so M2 is twice, okay, M2 is twice of M1, okay. So, or you can write here it as M, and you can write here, okay, this as 2 into M. So now you can write position of the center of mass, okay. This is M1 x1 plus M2 x2. So M into x1, so 0. 2m into x2, which is 10r, okay. So 2m into 10r divided by m1 plus m2 if you do. So that m1 plus m2 is actually 3m. Okay, m and m cancelled out. So this is the center of mass position, which will actually remain same. Okay, so when they, okay, here is the center of mass position, okay, somewhere here. The center of mass position is not going to remain change. Okay, now what you can do actually, so these are the two spheres, okay, in contact, okay, they are in contact, and here is the center of mass position, right? So what will happen, they will collide at a point. The point of collision I want to find out. So that point of collision, okay, will be such that center of mass position is not going to change. Okay, so center of mass position it is at just 20 r by 3. Okay, it is at just 20 r by 3. Or let me take that it is at 0. Okay, it is at 0. So you can write the new center of mass position xcm dash, that is m1 x1 dash plus m2 x2 dash divided by m1 plus m2. Suppose the new positions are x1 dash and x2 dash. Okay, x1 dash and x2 dash. Okay, now if I take the origin at x1, okay, origin at the at the center of the first one. So this is 0. From the center of, okay, from the center when they are in contact, from the center of the first one, the center of the second one, okay, because they both have radius r, that is at 2r, right? So you can have here, okay, m1 is m, so you can write 2m into 2r divided by 3m. M and m cancelled out. So 4r by 3. This is the position of center of mass. Okay. If, okay, this is the position of center of mass. If I take the origin here, okay, if I take the origin here, okay. Now you know that okay, this center of mass remains at the same position, but from the new position of the center of the first, it is at a distance 4r by 3 towards right. Okay, 4r by 3. Okay, so you know that this distance is r here. Okay, this distance is r. That means center of mass is at a distance r by 3 from the center of it. Okay, from the, okay, from this point of contact, the distance of the center of mass will be r by 3. Okay, just one second. This is r. Okay, I'm measuring the center of mass actually from the center of the first one, from the new center of the first one, which is 4r by 3. Okay, so this is the location actually, right? So now the location of the contact point is this here. Okay, here, this is the tangent. So I can subtract r by 3. Okay, r plus r by 3 is 4r by 3. I can subtract r by 3 from this 20r by 3. So if I subtract that, what I'm going to get? I'm going to get 20r by 3. That is the initial condition. Okay, initial position. This is r by 3. So you can write 20r by 3. Okay, 20 R by 3. Let me check once more. Okay, two uniform non-conducting identical size made up of different density. Density of A is twice the density of B. Okay, so mass of M2 is not 2M1. Okay, this is, here is the mistake I made. M2 is M if then M1 is equal to 2M. Okay, then you should take here as M only. Okay, so M into 10R. Now you can get 10R divided by 3. Okay, this is the position of center of mass actually. Okay, now the position of the center of mass after the collision from the center of the first one. Okay, this is the center of the first one. Okay, so here is the contact point. Okay, so xcm dash, that comes out to be, now in place of M2 you again need to take only M, not 2M. Okay, so M into 2R divided by 3M. Okay, so M and M cancelled out, you are now left with 2R by 3. Okay, 2R by 3. Now the distance of the contact point will be R from the center of the first one, but the center of mass is at a distance 2R by 3. Okay, 2R by 3. What does that mean? Okay, so from the new position, okay, x1 dash, okay, this is the new position and this is x2 dash, center of mass is at a distance 2R by 3, x equal to 2R by 3. Okay, or let me write this position as x dash. Okay, 2R by 3. Now the contact point is at a distance R, so here will be somewhere the contact point. Okay, suppose C is the contact point. Now the distance of the contact point, okay, will be equal to, if I make a subtraction, okay, the initial position was 20, this 10R by 3. So if from this 10R by 3 I subtract, okay, so this is R by 3, actually this is R by 3. Okay, 1 second, we have taken this, okay, twice M, 3M, 2R by 3. Okay, 2R by 3. Then okay, this is also okay, so contact will be at 10R by 3 minus R by 3. Okay, if I do that, it must be equal to 3R. Okay, so something, okay, just 1 second, let me take this question number 169 first. So a projectile of mass m is launched with an initial velocity v1 vector making an angle theta with the horizontal as shown in figure. The projectile moves in the gravitational field of the earth. The angular momentum of the projectile about the origin when it is at the highest point. So at the highest point, velocity is just equal to v1 cos of theta. v1 cos of theta. This is the height, okay, from the ground, and it will give me the perpendicular distance between them. This height is equal to, this height is v1 square sine square theta divided by 2g. You multiply both of them and you are going to get the angular momentum value, which is, okay, v1 okay, cube sine square theta cos theta divided by 2g with m. Okay, m into v into r perpendicular we are taking. Okay, if you apply the dot product, okay, so velocity is in the x direction at the top point. Okay, and you can see, okay, you can just take this r cross p. If you take that r cross p, it comes out to be minus k cap. Okay, so into the plane, that will be, first option will be the correct one. Now there are three liquids, A, B and C have same specific heat and mass m. 2m and 3m have temperature 20 degrees Celsius, 40 degrees Celsius and 60 degrees Celsius respectively. Temperature of the mixture when A and B are mixed. Okay, so when A and B are mixed, what will be the temperature if A and B are mixed? So you can write A for A and B, for A mass is m, for B mass is 2m. So you can write, suppose for A and B, for A, suppose temperature is 20 degrees Celsius. For B, suppose temperature is 40 degrees Celsius. Suppose mixture is having a temperature T. So mass of, okay, m into S into T minus 20 is equal to twice of m into 40 minus t. If you solve it, m and m cancelled out. Okay, s and s cancelled out. Okay, t minus 20 is equal to 80 minus 2t, 3t. Okay, what you are going to get? This 3t is equal to 100 and t is equal to 33 degrees Celsius. So 33 degrees Celsius, that will be for A. For A, the right match is fifth one. For B, if A and C are mixed, if A and C are mixed, A is going to have, A is having a mass m, C is having a mass of 3 m. Okay, specific heats are same. For C mass is, for C temperature is 60 degree, for A it is 20 degree. Okay, so you can write m into T m into S into T minus 20, that is equal to 3 into m. Okay, this is for A and C I am writing. 3m into S, then you can have 60 minus T. Okay, 60 minus T. Now m cancel, S cancel, m, S cancel. T minus 20 is equal to 180 minus 3t. 4t is equal to 200, that t is equal to 50. So for this 50, okay, you will get 50. Okay, 50 degrees Celsius. Then for the, okay, so for B, the third one. For C, B and C are mixed. Okay, if you are mixing B and C. So 2m into let me take here only. Okay, 2m into S into T minus 40, that I am writing for B and C. 3m into S into 60 minus T, ms, 2t minus 80 equal to 180 minus 3t. Okay, this is 260 equal to 5t. T comes out to be 52 degree. Okay, so 52 degree, that is for this C. That is for the C. D, A, B and C all are mixed. If you mix all of them, so first you take the mixture of A and B, then you can take up that mixture with C. So A and B is going to have a temperature of 33 degrees Celsius. Masses are m and 2m. So for that mixture you can write 3m. Okay, 3m into S into that T minus 20, into that 30, T minus 33. Okay, C is having greater temperature than the mixture of A and B. So heat will be absorbed by A and B. So T minus 33, that comes out to be equal to 3m into S into the 60 minus T. So 3m and 3m cancelled out, S and S. Okay, 60 minus T is equal to T minus 33. So you are going to get 2t is equal to 93 and t is equal to 47 around. Okay, so fourth option. Okay, that fourth option will be for D. Okay, fourth option will be for D. So for A, fifth is the correct match. For B, there is third. For C, it is second. And for D, this is fourth. So right option is the second one. Next one. Okay, next question. It takes 10 minutes to cool a liquid from 61 degrees Celsius to 59 degrees Celsius. If room temperature is 30 degrees Celsius, then find the time taken in cooling from 51 to 49. So I'm just going to use that law. Okay, Newton's law of cooling. Theta 1 minus theta 2 divided by T is equal to a constant, suppose K. Then average of theta 1 to theta 2 minus average of minus temperature of the surroundings. Now it is taking 10 minutes to cool a liquid from 61 to 59. So 2 divided by that T, okay, let me call it T1, it is equal to K, theta 1 plus theta 2, so 61 plus 59. 61 plus 59 divided by 2 minus 30. Okay, 61 plus 59. This comes out to be, this average will give me 60. So 2 divided by 10 is equal to k into 60 minus 30. 60 minus 30 is 30. So k divided by 2 divided by 10 divided by 30. It is 1 divided by 150. Okay, 1 divided by 150 minute inverse. Okay, that is the constant. Now theta 1 minus theta 2. You use this 51 minus 49. So 51 minus 49 divided by 2, it comes out to be k into 51 plus 49 by 2. Okay, that is 50 only. 50 minus theta naught. That theta naught is equal to, okay, this theta naught is equal to 30. So 20. Okay, 51 minus 49. Okay, 51 minus 49 divided by the time taken. So 2 divided by the time taken is equal to 150 into 20. Okay, 0, 0, 2 and 2. So this t comes out to be 15 minutes. Right answer is 15 minutes. Figure shows the pressure P versus volume V graphs for a certain mass of a gas at two constant temperature T1 and T2. Which of the following references is correct? Okay, so you can now write PV equal to nRT. Okay, so PV is equal to nRT1 at a temperature T1, PV equal to nRT2 at a temperature T2. For T2 greater than T1, okay, so this curve will remain above it. Okay, so from this diagram we can estimate T2 is greater than T1. T2 is greater than T1. PV graph for an ideal gas undergoing polytropic process, PV raised to the power m. So you can write PV raised to the power m is a constant. If you take derivative of both sides, this is m into p into v raised to the power m minus 1 dv plus dp into v raised to the power m is equal to 0. Okay, and now you can write m into p into v raised to the power m minus 1. Okay, this is equal to minus of dp into v raised to the power m. Okay, and now you can write this dp. Okay, this is dv here. This dp by dv is equal to minus of m, okay, into p divided by v. Okay, minus of m into p divided by v. Now if you see the value of, okay, you can now see m, they want the value of m. Slope is 1037 degree. Okay, 1037 degree is 3 by 5. So this 3 by 5 is equal to, okay, now I can write here 3 by 5 is actually equal to p by m into p by v. So p by v, if you do, you will get 1 by 2 only. Okay, so m into 1 by 2. So m comes out to be 6 by 5, which is 1.2. Okay, 6 by 5 is there? No. Okay, okay. So m into p by v we are having. Now this p by v, okay, at this point p is 2 into 10 raised to the power 5, v is 4 into 10 raised to the power 5, 1037 degree, sine 37 degree is 3 by 5, 3 by 4, not 3 by 5, it will be 3 by 4, 3 by 4. So 3 by 2 will be the right answer for this question. Okay, 3 by 2. Now, a uniform elastic rod of cross-section area A, natural length L and Young's modulus Y is based on a horizontal surface. Now, two horizontal forces of magnitude F and 3F directed along the length of rod and in opposite direction act at two of its ends as shown. After the rod has acquired steady state, the extension of the rod will be. Okay, so you can write extension of the rod delta L is equal to net force L divided by A divided by Y. Now that net force is 3F minus F, which is 2F. So 2F into L divided by AY. First option is the correct one. Two mercury drops, each of radius is small r, merged to form a bigger drop. If sigma is the surface tension of mercury, then surface charge, then surface energy released is. Okay, what that surface energy released is? So you can write the surface energy released. Two small mercury drops, they give a bigger drop. Okay, so you can write 4 pi by 3 r cube plus 4 pi by 3 r cube. It comes out to be 4 pi by 3 into capital R cube. Okay, So this 2 into small r cube is equal to capital R cube. Okay, and now you can write this capital R is equal to 2 raised to the power 1 by 3 into small r. 2 raised to the power 1 by 3 into small r. Now you can write surface energy. Okay, in the first case, it is sigma into 4 pi into small r square. In the second case, when there is a bigger drop, it is equal to sigma into 4 pi into capital R square. If you make a square of it, okay, so sigma into 4 pi into 2 raised to the power 2 by 3 into r square. Okay, and now if you take the difference, okay, 2 mercury drops were there, so earlier that energy is 8 pi times of r square. Okay, this is only for single one. Okay, and if you take the difference, so the right answer is actually the first one. Now a rectangular metal plate has dimensions of 10 centimeter into 20 centimeter. A thin film of oil separates the plate from a fixed horizontal surface. The separation between the rectangular plate and the horizontal surface is 0.2 millimeter. An ideal string is attached to the plate and passes over the ideal pulley to a mass m. When m is equal to 125 gram, the metal plate moves at constant speed of 5 centimeter per second across the horizontal surface. Then the coefficient of viscosity of oil in dynes per centimeter is. Okay, how we are going to solve it? You can just write okay, F is equal to eta into okay, or this eta, let me write eta directly, that is F by A. Eta is equal to F by A divided by this delta V divided by Z. If you see this force, that is actually the tension, and the tension is 125 gram into G. Okay, 125 gram into G, G is 1000. Okay, so 125 into 1000. If I do so, 125 into 10 raised to the power 3. 125 into 10 raised to the power 3. You divide it by the area, which is also in centimeter, 10 into 10. So 200. So 2 into 10 raised to the power 2. Velocity, okay, if you see the velocity, okay, velocity, they have given 5 centimeter per second. When we are going to depth, it becomes 0. So delta V is 5 centimeter per second. Okay, 5 centimeter per second. You take this, okay, thin film, okay, this jet to be 0.2 millimeter. So if you take this to be 0.2 millimeter, 0.2 into, okay, 0.2 into 10 raised to the power minus 1, that is 0.02. So if you calculate it, okay, you will get the answer is 2.5. Okay, do the calculation and you will get this answer. Now question number 177. A simple pendulum of length 1 meter is allowed to oscillate with amplitude 2 degree. It collides elastically with a wall inclined at 1 degree to the vertical. So a very small displacement it is having. Okay, so you can actually use this equation. Theta is equal to, for this question, theta is equal to theta naught sine of omega t. So when amplitude is half, what is the time, okay, in moving from here to here? If you see the length of it, then the time period for this for the simple pendulum is 2 pi into square root of L by G. L is 1 meter, so 2 pi into square root of 1. In place of G, you can use this pi square, you take it out, so you have 2 seconds. Okay, now in moving from here to here, this is only 1 second. In moving to here, from here to here, how much it is? Okay, so in moving from here to here, okay, how I can use this? Theta equal to theta naught sine omega t. Okay, so if you write theta naught by 2, that is equal to theta naught into sine omega t. Theta naught and theta naught cancel out, sine omega t, that is equal to pi by 6. Now you can write 2 pi divided by capital T into small t is equal to pi by 6. Okay, pi and pi cancel out, this is small t is equal to capital T divided by 12. That is 2 by 12, that is 1 second, 1 by 6 second. That is the time in moving from mean position to one of the extreme position. Okay, so this is 1 by 6. This is 1 by, okay, 2 by 4 that will be, that is 1 by 2 second. So the time period for this one is 1 by 2 plus 1 by 6 into 2. Okay, so this comes out to be 1 plus 1 by 3 and it is equal to 4 by 3 second. 178, a wire of length L having tension T and radius R vibrates with fundamental frequency F. Another wire of the same material with length 2L having tension 2T and radius 2R will vibrate with fundamental frequency. So the mass of the first one, if you write, mass of the first one is equal to density of it into volume of it. Mass of the second one, if you write m dash, that will be density, okay, into volume, pi r square L here. So if you have 4r square into 2L, so 8 rho pi r square L, 8 rho pi r square L. M dash, let me write, this will be 8 rho pi r square L. If you write the mass per unit length in the first case, okay, suppose that is mu. In the second case, if it is mu dash, then mu dash, you divide this mass 8 rho pi r square L by its length, you are going to get 4 pi r square L. So m dash is actually 4 times, mu dash is 4 times of mu. Okay, if you write the frequency in the first case, that frequency in the first case is 1 divided by 2L into square root of T by mu. In the second case, it is 1 by 4L because length is doubled now. Square root of T divided by 4 mu. Square root of 4, that is 2. So you can take a 2 outside, you can have 1 by 8L. Okay. So you solve it and you are going to get your answer as the third one. Maximum height reached by a rocket fired with a speed equal to 50% of the escape velocity. Okay. If you calculate that, you can write 1 by 2 mv square, 1 by 2 mv square, mve square plus g into m into small m divided by r. With a negative sign you can take it, with a negative sign you can take it, and it is equal to, at the point of maximum height kinetic energy is 0, then minus g into capital M into small m divided by r plus h. Okay, half of the speed, they are taking half of the speed, half of the escape speed. So this v dash is actually equal to half of the escape speed, which is square root of 2gm divided by r. If you make a square of it, you are going to get 1 divided by 2 into small m, okay, square of it, that will give me 1 by 4, then 2 into g into m divided by r minus g into capital M into small m divided by r. This comes out to be minus g into small m into capital M divided by r plus h. You can see this 2, 4, okay, this 2 and 2 cancelled out. We are left with minus of 3 into capital G into capital M into small m divided by r, that is minus g into capital M into small m divided by r plus h. Cancelled out, okay. You are now can write 3r plus h is equal to r, okay. So 3r plus 3h, that is equal to r. So 3h is equal to 2r, h is equal to 2r by 3, okay. H is equal to 2r by 3. So let us see, okay, I think, okay, let me see this, 1 by 2 mv dash square, 2r by 3, no, r by 3, that will be the, 1 second, 1 by 2 mv dash square, so 1 by 2 into square root of 2 gmr, make a square of it, okay, 1 by 2 into m, square of it, that is 1 by 4 into 2 into gm by r, 2 and 2 cancel out, so gm into capital M, okay, 1 by 4 here, okay, that is plus, this is minus 1, so minus 3 by 4, okay, minus 3 by 4 you will get, okay, so now 3r plus 3h, that is 4r, okay, and from this I can write r is equal to 3h, h is equal to r by 3, okay, that will be the answer, r by 3. Next one, a satellite is moved from one circular orbit around the earth to another of lesser radius. What is the following, okay, which one of the following statement is true? So lesser radius, if you remember the orbital velocity, okay, there is a relation between orbital velocity and, so mv square by r, it is equal to g into capital M into small m divided by r, small m and small m cancelled out. V orbital velocity is equal to G into capital M. Okay, this is R square divided by R. Okay, so this is square root of G into capital M divided by small r. Okay, this is the orbital velocity. Now lesser radius means more orbital velocity is there. Okay, kinetic energy, if I write kinetic energy, that is 1 by 2 into small m into V naught square G into capital M divided by R. So G into capital M into small m divided by 2R, this is the kinetic energy. Okay, so if radius is decreasing, kinetic energy is increasing. Okay, so the kinetic energy of satellite increases. What about the gravitational potential energy? So gravitational potential energy, that is just minus of G into capital M into small m divided by small r. So if it is decreasing, potential energy is, magnitude of potential energy is increasing, but potential energy is decreasing basically. So the kinetic energy of satellite increases and the gravitational potential of satellite Earth system, okay, the kinetic energy of satellite increases and the potential energy of satellite Earth system decreases. That will be the answer. Second option, this will be the correct one. Okay, now let us move to that question. Okay, here. Okay, this is A, this is B. B is more dense. So center of mass will actually towards B. Okay, not towards A. This will be actually towards B. At a distance 10 r by 3. Okay, at a distance it will remain at a distance 10 r by 3. Okay, so you need to add r by 3 into it. It will be inside B, not inside A. Okay, so it will be r by 3 distance from this contact point. So r by 3 plus 10 r by 3, that will give you an answer 11 r by 3. Okay, so 11 r by 3, that will be the answer for this question. Okay, then this is all for this solution. Yeah, thank you everyone.